The ROV camera finds the edge of the pool before it finds the pool itself. The light moves over gray sediment, then over a raised rim of pale crust and mussel shells, and then the seafloor suddenly changes into something that looks like a shoreline. Beyond that line, the water is darker, smoother, and heavier, as if a small inland sea has been placed on the ocean bottom and left there without wind. A crab walks along the margin and stops with its legs spread against the slope. Particles drift down through the beam, but when they touch the brine's surface, they hesitate and spread sideways, tracing a boundary that should not exist inside the ocean. The machine is already under hundreds of meters of seawater, surrounded by pressure and cold. Yet in front of it is another body of water with its own surface, its own edge, and its own danger. The first impression is simple enough to understand. It looks like a lake. But the word lake does not fully explain what the camera is seeing, because this lake has no air above it, no waves from weather, no sunlight falling onto it, and no ordinary shoreline where plants root and insects move. It is a lake under the sea, made not by rain or rivers, but by salt, pressure, chemistry, and the slow movement of buried layers beneath the seafloor. The title makes it sound frozen, and in one sense that word is useful, because almost everything about this place seems locked in place. The brine holds still. The animals respect the edge. Dead bodies can remain in the anoxic water far longer than they would in ordinary seawater. But in another sense, the word is misleading, because most deep-sea brine pools are not frozen solid. They are liquid, dense, hypersaline basins, sometimes cold, sometimes warmer than the surrounding bottom water, and sometimes surrounded by the separate kind of deep-sea ice known as methane hydrate. If places like this are why the hidden ocean keeps pulling at your curiosity, subscribing keeps the next descent within reach. The pool remains in the light, and the camera keeps moving, because the only way to understand it is to approach slowly, through the animals at its edge, through the chemistry below them, and through the old geology that made this strange surface possible. Normal seawater is already salty enough to sting your eyes and dry on your skin. A brine pool takes that condition much farther. In many deep-sea pools, the brine can be several times saltier than the water above it, and that extra salt gives the fluid extra density, enough density that it can sit on the seafloor like a separate layer and resist mixing with the ocean that surrounds it. Density is the quiet force that gives the pool its shape. It is not a wall in the solid sense, and an animal can cross it with a body or a claw, but it is still a barrier. Water above the pool contains oxygen. Water inside the pool may contain little or none. Water above the pool has a salinity close to the open ocean. Water inside can be so concentrated that most animals entering it cannot keep their cells functioning long enough to escape. The boundary is called an interface, and in ROV footage it can look like a sheet of glass, a layer of oil, or heat shimmer rising from pavement. Lights from the vehicle bend slightly as they pass through it. Sediment falling from above spreads along it. When the ROV thrusters disturb the water, the interface ripples with slow waves that travel across the brine surface. And for a moment, the seafloor lake behaves almost like a pond in wind. That visual beauty is part of why brine pools are so often described as mysterious. But the mystery is not supernatural. It is fluid physics and chemistry made visible. Two waters with different densities are refusing to mix quickly. Salt has changed the behavior of the ocean at a local scale, drawing a line across the seabed that small animals can find, avoid, exploit, and sometimes cross by mistake. The deep sea is often imagined as one continuous environment, but it is divided by layers and boundaries. Light fades with depth. Temperature changes with water masses. Oxygen can rise or fall depending on circulation and biology. Food descends unevenly as marine snow, carcasses, mucus houses, sinking fecal pellets, and the remains of plankton blooms. A brine pool adds another boundary to this already layered world, creating a place where the bottom is not simply bottom, but a meeting between one ocean and another. To reach many of these pools, a vehicle must descend through the upper ocean, where sunlight still shapes the day. Near the surface, plankton drift in water that changes quickly with weather and season. Farther down, the blue grows darker, and red light disappears. Animals in the twilight zone begin to carry their own light, and each flash can serve as signal, lure, camouflage, or warning in a water column where vision has become a narrow and uncertain sense. Below the twilight zone, the camera enters a darker volume where the surface world is present mostly as falling debris. Marine snow passes the lens in tiny white flecks. A lanternfish might flicker outside the beam. A squid may turn once and disappear. The descent is not empty, but it is sparse at the scale of a camera, and every living thing there is shaped by the cost of energy in a habitat where food arrives slowly, unless something unusual concentrates it. Cold seeps are one of those unusual concentrations. They are places where methane, hydrogen sulfide, oil, brine, or other chemically rich fluids escape from cracks and pathways in the seabed. They are called cold not because they are always icy, but because their fluids are usually close to the temperature of the surrounding seawater, unlike the superheated water released at hydrothermal vents. The name separates them from vents, but it does not make them simple. At a seep, the ocean floor is not just receiving food from above, it is receiving chemical energy from below. Microbes can use methane or sulfide in ways that animals cannot, converting chemical gradients into living tissue. Those microbes may form mats on the sediment, live inside the tissues of mussels or tube worms, or operate within thin layers of water where oxygen from above meets reduced chemicals from below. A brine pool is often part of this seep world, but with an extra complication. The brine itself may be moving upward after dissolving buried salt. It may be rich in methane. It may carry sulfide. It may be so dense that it flows downhill along the seabed, like a river with no surface exposed to air. Where it settles in a depression, the camera sees a lake. Where it slides along a slope, it can appear as a dark ribbon moving over sediment. The story of many Gulf of Mexico brine pools begins long before the modern Gulf had its present shape. During the Jurassic, when the Atlantic was opening and continents were shifting apart, shallow seas and restricted basins left behind thick deposits of salt. Later, sediments buried those deposits under layers of mud, sand, and organic material. Over millions of years, pressure and heat changed that buried landscape into a complex, deep structure of salt bodies, faults, domes, and pathways. Salt behaves strangely under pressure. It is solid, but over geological time, it can move like a very slow fluid, rising and deforming under the weight of sediments. In the Gulf, those buried salt layers helped shape the seafloor above them. They created traps for oil and gas, pathways for fluids, and places where seawater or formation water could dissolve salt and become concentrated brine before migrating upward through cracks and weaknesses. When the brine reaches the seabed, it does not spread evenly into the ocean like dye in a glass. It is too dense for that. It moves downward along the path of least resistance, fills hollows, spills over low points, and sometimes builds raised rims of minerals, shells, and carbonate crust. The result can be a pool with a margin so distinct that animals appear to gather along it in a ring. ROV footage from the Gulf often shows that ring before anything else. Mussel shells crowd the slope. Tube worms stand like pale tubes from the sediment. White bacterial mats coat patches of seafloor where chemistry reaches the surface. Fish hover nearby, not inside the brine but close enough to use the structure and the animals around it. The pool is lethal to many organisms, yet its edge can be crowded with life. That contradiction is central to the habitat. The brine interior can be hostile, anoxic, hypersaline, chemically harsh, and difficult for most animals to survive. The edge can be productive because it is exactly where different worlds meet. Oxygenated seawater brushes against reduced chemicals. Microbes use that boundary. Animals use the microbes. Predators and scavengers use the animals. A thin line between danger and nourishment becomes an ecosystem. Mussels at these seeps are not ordinary shoreline mussels displaced into deep water. Many belong to seep-associated groups that depend on symbiotic bacteria living in their gills. The bacteria use methane or sulfide as an energy source and provide nutrition to the mussel. The mussel provides a protected place for the bacteria and access to the chemical gradient. Together they make a living where sunlight cannot support plant growth. A single mussel bed can be dense enough that the shells form a visible pavement on the seafloor. Between them, small crustaceans move through gaps. Polychaete worms occupy tubes and crevices. Snails graze on microbial films. Brittle stars extend arms into the current. The animals are not there because the brine itself is gentle. They are there because the edge of the brine provides access to chemistry that can be turned into food. Tube worms at cold seeps follow a related logic. They have no ordinary mouth or gut as adults in some groups. Instead, they house bacteria inside specialized tissue, and those bacteria use chemicals from the seep to produce organic matter. The worm extends a plume into the water, where it can exchange gases, while its buried end reaches into sediment chemistry. It is an animal built around a partnership with microbes. The long lives of some seep tube worms show how stable these chemical habitats can be. A seep may not be eternal, but it can last long enough for slow-growing animals to settle, build tissue, reproduce, and create habitat for others. In a deep sea where a whale fall may feed scavengers intensely but temporarily, and marine snow arrives grain by grain, a seep can become a long-duration island of chemical productivity. Still, the stability is never complete. Brine flow can shift. Sediment can slump. A pool edge can move and drown a mussel bed that once marked the safe margin. Carbonate crust can grow, break, or be buried. A seep community may appear fixed in a single rough pass, but over years and decades it can change as the plumbing under the seafloor changes. The camera does not see that slow change unless people return. One dive shows the surface. Another dive years later shows whether the mussel rim has advanced or died back, whether a new mat has appeared, whether the brine has overflowed, whether a patch of sediment has been buried by fluidized mud. The deep ocean keeps its records, but they are not always easy to read from one visit. Some brine pools have become known because the footage is so visually clear. The pool called the Hot Tub of Despair in the Gulf of Mexico is one example, famous because the ROV view shows a circular brine pool with a distinct rim, and because the water inside was warmer and saltier than the surrounding deep sea. Its name is dramatic, but the science behind it is direct. Brine more than 4 times saltier than seawater, dense enough to pool, harsh enough to kill many animals that fall in. A pool like that can look calm, even when it is chemically severe. The surface may ripple softly under the ROV lights. The edge may be covered with animals. The water column above may be cold and dark but breathable for deep-sea fish. Just centimeters below the interface, the chemistry changes into something that those fish cannot tolerate. The difference between living and dying can be the thickness of a body length. When a fish crosses into an anoxic brine pool, the danger is not a predator with jaws or a sudden impact. It is the failure of physiology. The gills that normally extract oxygen from seawater encounter water with little oxygen and extreme salinity. The balance of salts across membranes is disrupted. The animal may struggle, lose coordination, sink, and become part of the pool bottom. That is why some brine pools contain preserved bodies. Fish, crabs, amphipods, or other animals that enter and cannot escape may settle into an environment where ordinary decomposition is slowed by lack of oxygen and harsh chemistry. The pool can become a quiet trap, not because it reaches out, but because it is difficult for aerobic animals to survive once they have crossed far enough beyond the edge. The sight can be unsettling. A crab lying intact in dark brine does not look like a dramatic death. It looks paused. A fish may rest on the bottom, with its shape still recognizable. In ordinary oxygenated water, scavengers, bacteria, and currents would quickly break down the body. Inside some brine pools, the process is slowed, and the pool becomes both habitat boundary and preservational environment. Scientists are interested in that preservation for several reasons. It tells them about the chemistry of the pool. It shows how strongly the brine resists mixing with oxygenated seawater. In some places, especially deep hypersaline anoxic basins in the Red Sea and Mediterranean, sediments beneath brine can preserve fine layers with little disturbance from burrowing animals, creating records of environmental change that might be blurred or destroyed elsewhere. The Red Sea contains some of the most extreme brine systems known. These deep hypersaline anoxic basins can have sharp chemical layers, high salinity, low oxygen, and microbial communities adapted to conditions that would kill many familiar organisms. Some are warmer than surrounding waters. Some contain high concentrations of metals or sulfide. Each basin has its own chemistry, and each chemistry selects for its own microbial world. Calling these places lifeless would be a mistake. Large animals may avoid or die within the most extreme brine, but microbes can occupy the interface and sometimes the brine itself. Halophilic archaea, sulfate reducers, methanogens, and other microorganisms can live in chemical conditions that seem severe only because animals like us are built for a narrow range of oxygen, salt, and temperature. Microbial life at these interfaces is difficult to see in ordinary footage. A white mat can show where bacteria are dense enough to become visible. A reddish or colored layer may hint at microbes suspended near the boundary, but much of the diversity is molecular, revealed by water samples, genetic sequencing, incubation experiments, chemical measurements, and microscopy back in the laboratory. The ROV arm reaches into this world carefully. A sampler lowered too quickly can disturb the interface and mix layers that scientists are trying to keep separate. A push core entering sediment may collect a record of microbes and chemistry, but the moment it is brought upward, pressure and temperature begin to change. The sample is evidence, but also a transformed piece of the original habitat. Deep sea science often works under that compromise. Cameras preserve behavior, but only in the beam of artificial light. Samples preserve chemistry, but only after being removed from pressure, darkness, and exact temperature. Sonar maps terrain, but not smell, texture, or microbial metabolism. Every tool reveals something and changes something else, and the best picture comes from combining many partial views. This is why brine pools are usually understood through repeated kinds of evidence. Researchers map the seafloor to find depressions, mounds, faults, and flows. They measure salinity and temperature with sensors. They collect water from above, at the interface, and within the brine. They sample mussels, worms, sediments, and microbial mats. They return to video and still images, checking where animals were found in relation to the chemical boundary. The result is not a single discovery that explains everything. It is an accumulating pattern. Dense brine sits on the seafloor because salt has raised its density. In many places, methane and sulfide provide chemical energy. Oxygen disappears inside the brine because mixing is limited and microbes consume what is available. Animals gather at margins where the chemistry is useful but not immediately fatal. The word frozen returns in a different way when methane hydrate appears. Methane hydrate is not ordinary ice, but it looks enough like ice that it has often been called methane ice or gas hydrate. It forms when methane and water combine under high pressure and low temperature, creating a solid structure in which methane molecules are trapped within cages of water molecules. At some cold seeps, methane hydrate occurs within sediment or as exposed white or yellowish material on the seafloor. It can appear as slabs, nodules, mounds, or crusts, sometimes partially covered by sediment and bacterial growth. Under the right pressure and temperature, it remains solid. Bring it to lower pressure or warmer conditions, and it can destabilize, releasing methane gas. In the Gulf of Mexico, Exposed gas hydrate has supported its own strange observations, including worms found living on or within hydrate mounds. These animals are small, not monsters, and their exact nutritional relationships have required careful study. They may graze on microbes growing on the hydrate surface or live in close association with methane-based microbial communities. The important point is that even seafloor ice can become habitat. Hydrate gives the title a more literal frozen element, but it should not be confused with the brine pool itself. A brine pool is liquid hypersaline water. Hydrate is a solid methane water compound stable under deep sea conditions. They can occur in related seep systems because both involve methane, pressure, cold, and subsurface fluid flow, but they are different materials with different behavior. The distinction matters because deep sea language can blur quickly. A pool may look frozen because it is still. A hydrate mound may look like ice because it is solid. A brinacle under sea ice may actually be made of ice growing around a sinking plume of cold brine. All three involve salt, cold, and density, but they are not the same phenomenon. Under polar sea ice, seawater freezes in a way that excludes much of its salt. The ice becomes fresher than the water it formed from, and the rejected salt concentrates in brine pockets and channels. That cold brine can drain downward because it is both colder and saltier than the surrounding water. As it sinks, it can freeze the seawater around it, forming a hollow tube of ice known as a brinacle. A brinacle can descend like a white finger from the underside of sea ice toward the seafloor in shallow polar water. When the cold brine reaches the bottom, it can spread along the seabed and harm slow-moving animals such as sea stars or urchins if they are caught in its path. This is one form of frozen brine in the ocean, but it belongs to a polar shallow water setting, not to the deep Gulf brine lakes formed by buried salt and seep chemistry. The deep sea has many ways to make boundaries out of water. Under ice, brine rejection can send dense water downward. In the Gulf, salt tectonics can create hypersaline fluids that rise and then sink again into depressions. At spreading centers and in enclosed basins, geothermally influenced brines may collect in deep hollows. The details differ, but the physical principle returns again and again. Salt changes density, and density organizes the ocean. For an animal, density is not an abstract number. It is something felt as movement, resistance, and position in the water. A fish hovering above a brine pool may experience a sharp change in buoyancy if it dips too low. A crab walking near the edge may touch the interface with one leg and pull back. A shrimp may use the structure of a seep without crossing into the harshest water. ROV pilots learn to respect the interface as well. Thrusters can push lighter seawater into the brine or lift brine upward in a cloud that temporarily obscures the camera. Sampling equipment can stir the boundary. Even the vehicle lights and arms, designed to observe and collect, become part of the scene. The machine is not invisible. It is a visitor with propellers, cables, hydraulic joints, and a bright artificial sun. Inside the control room, the pool is seen on monitors rather than through a window. The ship may roll gently at the surface, while the vehicle hovers in black water far below. Scientists call out observations. A pilot adjusts position by small increments. Numbers for depth, heading, temperature, and conductivity appear on screen. The place becomes known through a combination of image, instrument, and voice. Conductivity is especially important because it helps reveal salinity. Saltwater conducts electricity better than fresher water, and a sharp change in conductivity can mark where the vehicle has crossed into denser brine. Temperature sensors show whether the pool is colder, warmer, or close to ambient bottom water. Chemical sensors can detect methane, sulfide, oxygen, or pH changes. The lake on the screen becomes a set of gradients. Those gradients are the true architecture of the habitat. A rock can be seen directly, but a chemical boundary has to be inferred from water behavior, sensor readings, and biological patterns. Mussels cluster where methane and oxygen are both available in useful amounts. Bacterial mats appear where sulfide reaches oxygenated water. Dead animals collect where oxygen fails. The organisms draw a map of invisible chemistry. Food scarcity makes that chemical map even more important. Away from seeps. Much of the deep seafloor depends on material sinking from the surface. A bloom of phytoplankton may begin in sunlight, get eaten by zooplankton, broken into fecal pellets, repackaged into mucus, fragmented by small animals, and finally settle as a weak snowfall on mud thousands of meters below. By the time it arrives, much of its energy has been used. A cold seep changes the economy. It brings energy from the earth rather than from sunlight, not as warmth in the way a hydrothermal vent does, but as reduced chemicals that microbes can oxidize. Methane and sulfide become the beginning of a food web. The animals around a brine pool are still part of the ocean, still affected by currents and larvae and predators, but their local productivity is tied to subsurface chemistry. The edge of a brine pool can therefore resemble an oasis, but that word should be used carefully. It is not a garden in any gentle sense. It is a narrow zone where life is possible, because danger and opportunity sit close together. Step too far into the brine, and many animals die. Stay near the margin, and microbes can support dense communities that would be impossible on ordinary deep mud with the same amount of falling food. Fishes at seeps are often visitors rather than strict residents. They hover around structure, investigate disturbed sediment, and prey on smaller animals. Some species may use seep habitats regularly because the communities concentrate food and shelter. Others pass through as part of broader movements along slopes and basins. The camera records them as individuals, but their lives extend beyond the frame into darkness that the ROV never sees. Crabs and shrimp are more visibly connected to the bottom. They walk across carbonate crust, pick at mussel beds, feed on detritus, scavenge dead tissue, or graze on microbial films. Their claws and legs turn seep productivity into movement. They are not symbols of death around a toxic pool. They are animals solving ordinary problems in an extraordinary place, finding food, avoiding being eaten, reproducing, and staying within tolerable chemistry. Anemones and corals may appear on harder substrates near brine features where carbonate has precipitated from seep chemistry. These animals do not use chemosynthesis in the same direct way as symbiotic mussels, but they can take advantage of structure and currents. Hard bottom is rare in many deep sedimented areas, so any crust, rock, or shell accumulation can create attachment points that change the local community. Carbonate crusts form because seep chemistry alters the sediment-water environment. Microbial processes involving methane and sulfate can increase alkalinity and promote carbonate precipitation. Over time, this can build pavements, blocks, ledges, and rims. The brine pool shoreline is therefore not just a passive edge. It may be a mineral structure built partly by the chemistry and biology of the seep itself. The seafloor records these processes in texture. Smooth mud gives way to broken carbonate plates. Shell hash collects along slopes. White mats occupy soft patches. Darker brine fills low places. A laser scale on the ROV image gives size to the scene, reducing the strange landscape back into measurements, centimeters between mussels, meters across a pool, millimeters of bacterial film, degrees of slope along a rim. Scale matters because brine pools range widely. Some are small enough to resemble puddles or ponds on the seabed. Others are large enough to be mapped as basins. A depression filled with hypersaline water can be less than a meter across or stretch across areas large enough that an ROV cannot see the opposite edge in one frame. The word pool covers many forms. The largest systems change how people think about the seafloor. In deep basins of the Red Sea or Mediterranean, hypersaline anoxic water can fill broad depressions and create layered water bodies with distinct chemistry from top to bottom. These are not minor puddles. They are deep sea lakes in a more expansive sense, with interfaces, sediments, microbial communities, and histories of their own. Those histories can be surprisingly long. In an anoxic basin where burrowing animals cannot disturb sediment, thin layers may remain intact. Dust, plankton remains, chemical precipitates, and other particles settle year after year. A core taken from the bottom can preserve a sequence that helps reconstruct past climate, floods, earthquakes, or changes in surrounding environments. The pool becomes both ecosystem and archive, yet the archive is hard to access without altering it. A coring device must pass through the water column, through the interface, and into sediment. The recovered layers must be kept intact. Chemical gradients can shift as pressure changes. Microbes that were active under one set of conditions may slow, die, or change activity as the sample is handled. The scientific value is high, but so is the need for caution. This caution is one of the quiet themes of deep-sea research. The environment is difficult to reach, expensive to observe, and easy to disturb at small scales. A trawl net can bring up animals, but it often damages soft bodies and loses the position information that matters. A camera can preserve the scene, but it cannot identify every microbe or measure every chemical reaction. A sample can be analyzed in detail, but it is no longer exactly where it lived. Brine pools show why context matters. A mussel shell in a jar tells you that the animal existed. A tissue sample can reveal symbionts and isotopes, but the video of that mussel at the pool rim tells you about its position relative to the brine, the bacterial mat, the carbonate crust, the current, and the neighboring animals. Deep-sea evidence is strongest when the specimen and the scene remain connected. Museum collections still matter. Preserved mussels, worms, shrimp, fish, and cores allow researchers to return to material years later with new tools. DNA methods improve. Isotope techniques become more refined. Questions change. A specimen collected during one expedition may answer a question that had not yet been asked when it came aboard the ship. Environmental DNA adds another layer. Water collected near a brine pool may contain traces shed by animals and microbes, fragments of genetic material that reveal who has been present even when the camera did not see them. This is especially useful in dark, complex habitats where small organisms hide in sediment or shell beds. But eDNA also requires careful interpretation because currents can move traces and DNA does not always show whether an organism is alive, resident, or only recently passing through. The limits of evidence keep the narration honest. Scientists can say that many brine interiors are anoxic, that methane and sulfides support chemosynthetic communities, that mussels and tubeworms live at margins, and that some animals die when they enter the brine. They cannot say that every pool behaves the same way, or that one video captures the full life of the habitat. Each pool is a local expression of geology, chemistry, and biology. Even within one region, pools differ. A brine pool fed by active flow may have different chemistry from one that is more isolated. A pool rich in methane may support different microbes than one dominated by sulfide or metals. Temperature can vary. Salinity can vary. The thickness of the interface can vary. The animal community at the rim reflects those differences. Some pools are rimmed by mussels. Some are associated with tubeworm fields. Some have bacterial mats more prominent than large animals. Some lie near oil seeps where dark hydrocarbons stain sediment or coat surfaces. Others are linked to mud volcanoes where fluidized sediment escapes, building mounds and changing the seafloor abruptly. The category brine pool is useful, but it contains many individual stories. The Gulf of Mexico remains one of the best-known regions for these habitats because of its salt geology, hydrocarbon systems, and history of research. Scientific interest there has also been shaped by industry because oil, gas, salt structures, and seepage are connected. This creates a complicated human context. The same region that supports unusual deep-sea communities also holds resources that people have mapped, drilled, and extracted. Human activity does not erase the natural history of seeps, but it does make careful knowledge more important. Trawling can damage seafloor habitats. Drilling and infrastructure can alter local environments if not managed carefully. Oil spills can affect deep and surface systems in different ways. Baseline studies of seep communities help scientists understand what is natural, what is changing, and what may be vulnerable. The animals at brine pools did not evolve for human categories like resource, hazard, or spectacle. A mussel does not know that methane beneath it matters to energy markets. A shrimp does not know that a ROV video may be watched by millions. A tube worm does not know that its symbiotic bacteria interest microbiologists. These organisms are living within gradients, and their lives are measured by settlement, growth, feeding, reproduction, and death. Larvae are part of the story, even though they are rarely the focus of brine pool footage. Many seep animals must disperse through the water column before settling at a new habitat. A larva leaving one seep community enters a vast dark ocean where suitable chemical islands may be separated by kilometers or more. Finding the next seep is a biological challenge that shapes population genetics, distribution, and resilience. For sessile animals, settlement is a decision made by a body with limited information. A larva may respond to chemical cues, microbial films, currents, or substrate texture. If it settles too far from the seep chemistry, it may not receive enough energy. If it settles too close to harsh brine, it may die. The adult mussel bed that looks fixed and stable is built from many small, successful placements over time. Predators also shape the edge. Crabs can break shells. Fish can pick off smaller animals. Snails can graze. Starfish, where present, can move slowly through dense communities and feed in ways that alter local patterns. Even in a chemical ecosystem, ordinary ecological interactions continue. Chemosynthesis begins the food web, but it does not remove competition, predation, parasitism, or disease. The brine itself may protect some animals indirectly by setting a boundary predators hesitate to cross. A mussel at the right position might benefit from chemical access and from a dangerous moat nearby, but that benefit is conditional. If the pool expands, if brine overflows, if sediment slumps, the same boundary can become a threat. The habitat is productive because it is unstable at small scales. Footage of a brine pool can make the deep sea seem calm because the movements are slow. Mussels do not run. Tube worms do not chase. Bacterial mats do not visibly grow before the camera. But the chemistry is active. Methane diffuses. Sulfide reacts. Microbes divide. Carbonate precipitates. Brine creeps. Tiny animals feed and reproduce within shell beds. The still image is filled with processes. Pressure is another process made invisible by the image. At 1,000 meters, pressure is roughly 100 times greater than at the surface. At greater depths, it increases further, compressing gas spaces and affecting chemistry, materials, and biological membranes. The ROV is built to resist it with pressure housings, oil-filled cables, syntactic foam, thick glass spheres, and seals. The animals are not resisting pressure as an outside force in the same way. They are built to live within it. Bring many deep-sea animals to the surface, and they change. Gases expand. Tissues deform. Colors shift under sunlight. Soft bodies collapse without pressure and water support. A mussel shell may survive well, but a delicate worm or gelatinous animal may not. The deep sea often looks stranger in a trawl net than it did alive, because removal from habitat distorts the evidence. ROV footage helps correct that distortion. It shows animals in posture, not just as specimens. A fish hovering over sediment, a shrimp holding position in current, a crab stepping along a rim, a tube worm plume moving in weak flow, a mussel bed arranged along a chemical boundary. These details make the organisms less monstrous and more understandable because they reveal how each body works where it belongs. The pool also changes how darkness is understood. Darkness is not only mood, it is an ecological condition. Without sunlight, there are no plants at the brine pool edge, no algae growing like seaweed on rocks, no photosynthesis building the base of the local food web. Light from the ROV reveals the scene for human eyes, but it is not part of the natural energy system there. The energy comes from chemistry. Bioluminescence may still occur nearby, especially in the water column above slopes and basins. Small animals flash as they move through the dark. A fish may produce light from photophores. A shrimp may release a glowing cloud. But at the brine pool margin, much of the visible life is benthic, living on or in the bottom, and its energy is bound to microbes rather than to the flashes that punctuate midwater. The midwater and the seafloor are connected by falling material and migrating animals. Each night, many small animals rise towards surface waters to feed and descend again before daylight. Their waste and bodies carry carbon downward. Some become prey for deeper animals. Some die and sink. That background rain continues over brine pools, but it is supplemented by seep energy, making the pool edge more productive than surrounding mud. A dead whale on the seafloor creates another kind of deep-sea oasis. Scavengers arrive first, then bone-eating worms, sulfide-producing microbes, and animals using the chemical energy from decomposing skeleton. A brine pool differs because its energy source is geological rather than a carcass, but the comparison is useful. Both create concentrated habitats in a large dark environment where food is usually dispersed. Hydrothermal vents offer another comparison. Vents release hot, mineral-rich fluids from the crust, often at mid-ocean ridges or volcanic settings. They build chimneys, support chemosynthetic communities, and can change quickly. Cold seeps release cooler fluids and gases, often along continental margins, and may last longer in some places. Brine pools belong more naturally to the seep family, though some deep hypersaline systems have their own tectonic and geothermal histories. At a black smoker vent, the drama is vertical. Hot fluid rises, minerals precipitate, chimneys grow, shrimp or tube worms crowd near intense gradients. At a brine pool, the drama is horizontal. Dense fluid settles, spreads, and forms a surface. Animals arrange themselves along a shoreline. The strongest boundary may be a thin layer between waters rather than a plume rising into the ocean. Both habitats show that the deep sea is not merely a passive receiver of surface debris. The seafloor can feed life from below. It can release heat, methane, sulfide, metals, and salt. It can build structures. It can create hazards. It can support communities whose existence was once unexpected because people assumed sunlight was the only major route to biological productivity. The discovery of chemosynthetic ecosystems changed that view. Hydrothermal vents in the late 20th century showed dense animal communities thriving without sunlight. Cold seeps added a different version of the same principle. Brine pools complicate it further by placing extreme toxicity and productivity at the same edge. They are not exceptions to biology. They are evidence that biology follows usable energy wherever physical conditions allow. That does not mean anything can live anywhere. Limits remain real. Salinity can be too high for most animals. Oxygen can be too low. Sulfide can be toxic. Pressure and temperature constrain proteins and membranes. The animals at brine pool margins are not ignoring these limits. They are positioned precisely where their bodies and symbionts can stay within them. Microbes expand the apparent range of life because their metabolisms are so varied. Some tolerate high salt by balancing their internal chemistry with compatible solutes, or by adapting proteins to function in salty environments. Some live without oxygen by using other chemical pathways. Some use methane. Some reduce sulfate. Some oxidize sulfide. At the interface, different metabolisms can form layered communities, each using the products or conditions created by others. This layering can occur across millimeters. Oxygen from seawater penetrates only so far into microbial mats or sediments. Sulfide rises from below. Methane diffuses upward. Where they overlap, one group of microbes may thrive. Slightly deeper, another group takes over. Slightly higher, a different process dominates. The visible mat is a surface expression of a vertical chemical city. For animals with symbionts, the microbial world moves inside the body. A mussel filters water and maintains bacteria in its gills. A tubeworm supplies chemicals to bacteria housed in tissue. The animal becomes a controlled interface, managing the contact between oxygen and reduced chemicals in a way that feeds its internal partners. This is not a simple relationship, but it is one of the great themes of seep life. The partnership also creates vulnerability. If the chemistry changes, the symbionts may no longer receive what they need. If oxygen falls too low at the margin, the animal may suffer. If flow stops, a mussel bed may decline. If brine shifts over the community, the animals can be killed. Symbiosis solves one problem by tying the animal closely to a specific set of conditions. This is why seep communities can be patchy. A few meters can separate dense life from bare sediment. A slight rise in the seabed may keep an animal above the harshest brine. A small crack may deliver methane to one patch and not another. The ROV camera may turn from an empty-looking mud plain to a crowded mussel bed in seconds, not because the deep sea changes randomly, but because the chemistry is local. Mapping those patches requires patience. Multi-beam sonar can show mounds, depressions, and hard grounds. Backscatter can reveal differences in seafloor texture. Chemical sensors can detect anomalies. But the fine biological pattern often requires visual survey, moving slowly over the bottom, recording where animals appear and where they vanish. The map becomes biological only when the camera and samples confirm what the instruments suggest. Some of the most important evidence is almost ordinary. A temperature reading slightly above ambient, a conductivity spike, a dissolved oxygen drop, a cluster of shells, a black stain on sediment, a shimmering layer, a few bubbles rising from mud. In the deep sea, small signs can reveal large subsurface systems because the seafloor rarely presents its processes in a single clear gesture. Bubbles themselves behave differently at depth. Methane rising from the seafloor may dissolve in seawater, be consumed by microbes, or form hydrate skins under certain conditions. Much of the methane released at seeps may never reach the atmosphere because it is filtered through water column and sediment microbial processes. This microbial filter is important for understanding carbon cycling, even though its efficiency varies with place and conditions. The carbon in methane can be old, derived from buried organic matter transformed over geological time. When seep microbes use it, that old carbon enters modern food webs. A mussel growing at a brine pool edge may build tissue from carbon that began as ancient organic material trapped in sediment. A crab eating that mussel transfers the carbon again. The deep seafloor is part of the planet's carbon cycle, not separate from it. Sulfide adds another chemical path. It can be toxic to many animals because it interferes with respiration, but sulfide oxidizing bacteria can use it as an energy source. At seeps, sulfide may come from microbial sulfate reduction in sediments, often tied to methane oxidation. The same compound that makes one layer dangerous can help support life at another layer when bacteria place it within the right reaction. Salt does not provide energy in the same way methane or sulfide does, but it shapes the habitat by organizing fluids and excluding many organisms. High salinity can pull water out of cells, disrupt proteins, and make ordinary animal physiology fail. For microbes adapted to it, salt becomes part of their operating environment. For fish, crabs, and many worms, it is a boundary to avoid. A brine pool therefore has three overlapping identities. It is a geological feature made by salt movement and fluid flow. It is a chemical feature marked by salinity, oxygen loss, methane, sulfide, and sometimes metals. It is a biological feature where microbes and animals gather along gradients. None of those identities alone is enough. The pool is all of them at once. The visual language of a shoreline can help, but it can also mislead. On land, a shoreline separates water from air. Around a brine pool, the shoreline separates one water from another. There is no beach in the ordinary sense, no tide mark left by wind-driven waves, no plants rooted in damp sand. The rim is made of sediment, salt-influenced minerals, carbonate crust, shells, and living animals arranged around a dense liquid. ROV lights can make that rim look brighter than it is. White shells reflect strongly. Bacterial mats glow pale under artificial light. Carbonate crust appears chalky. In natural darkness, the scene would have no visible color unless an animal produced light or a human machine arrived. The brightness in footage is a temporary human condition imposed on a habitat that normally functions without it. The animals may still sense the ROV. Lights can startle fish or attract some organisms. Thrusters can disturb sediment. Sound and vibration move through water. A manipulator arm approaching a sample may cause a crab to step away. Observations are real, but they are observations of animals responding partly to a visitor. Scientists account for this by watching carefully and by comparing many encounters. One reason brine pools feel cinematic is that the camera gives them scale slowly. At first, the frame shows only texture, mud, shells, a dark surface. Then, the ROV moves and the edge curves away. A fish crosses the foreground, a laser dot shows distance. The pilot gains altitude, and suddenly the viewer understands that the pool extends beyond the frame, a dark body sitting under an already dark ocean. The same scene on a sonar map looks less emotional but more complete. A depression becomes a shape. A mound becomes a relief feature. Channels show where dense brine may have flowed. Hard returns suggest carbonate or shell accumulations. The sonar image cannot show the crab at the edge, but it can place the crab's world within a larger landscape of slopes, basins, faults, and seep structures. Continental slopes are especially important because they are neither shallow shelves nor flat abyssal plains. They are transitional regions where sediments accumulate, slump, fault, and channel. Organic matter from the shelf can move downslope through canyons. Hydrocarbons generated below can migrate upward. Salt bodies can deform the seabed. A brine pool on a slope is part of a dynamic margin, not an isolated curiosity. Submarine canyons can concentrate currents and food. Seamounts can alter flow and gather animals. Abyssal plains can stretch for enormous distances with relatively low food supply. Trenches impose great pressure and different sediment dynamics. Brine pools occupy their own places among these habitats, often associated with margins and basins where geology allows dense saline fluids to form and escape. The deep sea is often described by depth zones, but a brine pool reminds us that depth alone does not define a habitat. Two sites at the same depth can differ completely if one is ordinary mud and the other is a methane-rich brine seep. Pressure and darkness may be similar, but chemistry, substrate, food supply, and biological density can be radically different. For a fish moving along the bottom, the world may be a mosaic. There is soft sediment with scattered burrows. There is a carbonate ledge with attached animals. There is a mussel bed. There is a bacterial mat. There is a brine edge to avoid. There are currents carrying odor and particles. There are predators, prey, and places to shelter. The environment is not blank simply because the human eye sees darkness between illuminated patches. For a larval mussel drifting above the bottom, the mosaic is mostly invisible until chemical cues become strong enough. The larva cannot reason through geology, but it may respond to methane-associated microbes or other signals that indicate suitable habitat. Its success depends on landing in one of the rare places where its future symbionts and chemistry will support growth. For a microbe, the mosaic exists at a smaller scale. A grain of sediment, a shell surface, a mat layer, or a droplet of brine can be a habitat. Gradients that are invisible to a fish may define microbial life completely. Oxygen a fraction of a millimeter higher or lower can change which metabolism works. The brine pool is therefore vast or tiny, depending on the body experiencing it. This is one of the reasons deep sea life resists simple description. The same place can be lethal to one organism, nourishing to another, and merely a boundary to a third. A pool that kills fish can support microbes. A rim that threatens mussels if it shifts can feed them while stable. A hydrate mound that seems like frozen geology can host worms and bacteria. The deep sea is not kind or cruel. It is conditional. Scientific names can make the animals sound distant, but their bodies remain practical. Bathymodiolus mussels build shells, filter water, host bacteria, and anchor themselves in dense beds. Vestimentiferan tubeworms build tubes, extend plumes, and manage symbiosis. Polychaete worms crawl, burrow, or graze. Amphipods scavenge. Crabs test surfaces with legs and claws. Fish hover and strike when the chance appears. The ordinary behaviors matter because they prevent the habitat from becoming a fantasy. A brine pool is strange, but the animals around it are not performing strangeness for the camera. They are doing what animals do, exploiting resources, avoiding lethal conditions, competing for space, reacting to disturbance, and passing their genes into a next generation that must also find the edge and survive there. Reproduction is difficult to observe directly in many deep sea seep animals. Scientists infer life cycles from larvae, genetics, population structure, growth rings, seasonal patterns, and comparison with related species. Some details remain uncertain. How often larvae successfully move between seep sites, how long particular communities persist, and how quickly they recover after disturbance can vary across species and regions. Uncertainty does not weaken the story. It makes the habitat more realistic. The deep sea is not unknown because scientists have ignored it. It is difficult because each observation requires ships, weather windows, vehicles, funding, and instruments capable of working under pressure. Even then, the camera may arrive at the wrong season, the wrong time of day, or after a change that began years earlier. A single frame of a brine pool can suggest permanence, but the underlying system is always moving at some rate. Fluids migrate through sediment. Salt shifts under load. Methane is produced, trapped, released, consumed. Microbial communities respond. Carbonate grows. Animals settle and die. Sediment falls. The pool may remain recognizable for years, while every part of it changes slowly. Geological time and biological time overlap here. The salt that helps form Gulf brine began in ancient seas. The methane may come from organic matter buried long before humans existed. The mussel at the rim may live on a much shorter scale, perhaps years or decades. The microbe may divide far more quickly. The rough visit lasts hours. All these times meet in one illuminated patch of seafloor. That meeting of times is one reason the habitat feels larger than its size. A small pool can connect Jurassic salt deposition, modern microbial metabolism, deep sea animal ecology, and human technology in the same scene. The camera does not need to exaggerate the place. The facts are enough if they are allowed to remain connected. The title's frozen quality can also describe the way deep sea footage preserves a moment. A fish turns at the edge. A crab pauses. The brine surface ripples and smooths again. On the ship, the recording is saved with depth, date, location, and dive number. Years later, someone can watch the same moment and measure details the first observers missed. But the actual pool did not pause when the video ended. The RUV ascended. The ship moved on. Currents continued along the bottom. Bacteria kept using methane. Animals fed and died. The interface repaired itself after disturbance. The pool returned to darkness, not empty darkness, but darkness filled with chemical gradients and bodies adapted to them. The Arctic and Antarctic versions of dense brine add another chapter to the same broad physics, When sea ice forms, salt rejection helps create dense water that sinks and contributes to global circulation. On large scales, polar brine formation is part of how deep and bottom waters are produced, helping ventilate the ocean and move heat, oxygen, and carbon around the planet. On small scales beneath sea ice, cold brine can become visible as brinicles and bottom-hugging flows. These are more directly frozen than deep brine pools because ice grows around the descending brine plume. Yet even there, the core is brine, still liquid because salt lowers the freezing point. The ice tube is a structure around a moving chemical flow. The phrase frozen brine is therefore almost a paradox. Pure water freezes at a familiar temperature, but seawater freezes lower, and concentrated brine can remain liquid at temperatures that would freeze fresher water. Salt both comes from freezing and resists freezing. Ice rejects salt, salt creates brine, brine sinks, and the sinking brine can freeze water around it while staying liquid itself. In the deep sea, pressure adds another complication. Pressure can stabilize methane hydrate, allowing ice-like solids to exist at temperatures above the normal freezing point of seawater. The ocean bottom can therefore hold liquid brine and solid hydrate close together. One is made dense by salt, the other is held solid by pressure, cold, and gas. Both challenge surface expectations of what water and ice should do. A person standing on a beach experiences water in a narrow set of conditions: waves, air, sunlight, sand, salt, and weather. The ocean bottom has different rules. Pressure changes gas behavior. Darkness removes photosynthesis. Cold slows many processes. Salt layers create dense fluids. Methane becomes trapped in hydrate. Microbes use chemical pathways that surface animals never notice. The same planet contains both worlds. The surface ocean can hide this because it looks continuous. A calm Gulf surface gives no sign of salt lakes below. A polar ice sheet gives only a hint of brine channels draining beneath it. A research ship sees waves, weather, and instruments, while the seafloor holds pools, seeps, mounds, and animals arranged around invisible chemistry. The visible surface is not false, but it is incomplete. Descent makes the incompleteness obvious. The cable leaves the ship. The vehicle passes through blue water, then darker blue, then black. The temperature changes. Pressure rises. The ship noise fades into transmitted data and camera feed. By the time the ROV reaches the bottom, the surface world has become a set of support systems: power, control, navigation, and people watching screens. The brine pool does not need those people to exist. It formed without them and would continue without their camera. Human presence matters because it allows the scene to be known, not because it defines the scene. This is a useful humility in deep-sea exploration. The goal is not to conquer the bottom, but to understand a place that has its own processes, whether anyone is watching or not. Understanding also means resisting easy horror. The pool can kill animals, but it is not evil. It preserves bodies, but it is not a monster. It looks like a portal or a dark mirror, but it is a hypersaline fluid body shaped by geology. The real story is more interesting than a myth because it explains how a lethal interior can support a living rim. The same restraint applies to beauty. The pool is beautiful in ROV footage, but not because it was made for a human camera. It is beautiful because a physical boundary becomes visible, because animals arrange themselves around a chemistry line, because light catches the interface and turns density into image. The beauty is a byproduct of function. A scientist watching the monitor may feel awe, but awe does not replace measurement. The dive still needs samples, coordinates, sensor readings, and careful notes. The manipulator still has to close a bottle at the right depth. The vehicle still has to avoid stirring the pool before the interface is measured. Emotion can bring attention to the place, but evidence is what allows the place to be understood. The cold seep fact sheet version of the habitat is concise: methane, hydrogen sulfide, and hydrocarbon-rich fluids escape from the seafloor. Microbes digest these compounds. Brine seeps occur where seawater interacts with salt layers and becomes dense enough to form underwater lakes. The footage version is slower. A beam of light, a crab, a dark surface, a ring of shells, a hand-shaped manipulator entering the frame. Both versions are necessary. Without the concise explanation, the image remains only strange. Without the image, the explanation can become dry and detached from the living bottom. A good deep-sea story moves between them, returning to the camera whenever the chemistry becomes abstract and returning to the chemistry whenever the camera seems too magical. The brine pool is also a lesson in edges. Many ecosystems concentrate life at edges: shorelines, reef slopes, oxygen boundaries, upwelling fronts, ice margins, forest borders, river mouths. Edges bring difference into contact. At a brine pool, the difference is extreme, but the principle is familiar. Life gathers where gradients can be used. At the same time, not every edge is productive for every organism. The oxygen minimum zone can exclude some animals while supporting others adapted to low oxygen. A hydrothermal vent can cook one animal and feed another. A brine interface can be navigable for a crab but fatal if crossed too deeply. Habitat is not a universal condition. It is always habitat for someone. The animals that fail at the edge are part of the ecosystem too. A dead fish in brine may not be eaten quickly by ordinary scavengers, but it still represents energy, chemistry, and evidence. Its presence marks a boundary. Its preservation tells scientists about oxygen. Its body may eventually be altered by microbes suited to the pool. Even death becomes information. There is a quiet similarity between a brine pool and a fossil bed, though one is living and the other ancient. Both preserve traces under special conditions. Both require careful interpretation. Both can mislead if treated as complete records. A brine pool may preserve bodies that entered by mistake, not a full sample of the surrounding community. A fossil bed preserves what burial allowed, not every animal that lived. This connection to preservation fits the reference style of deep time, but the deep sea adds its own scale. Salt deposits from ancient seas rise into modern seafloor processes. Methane from buried carbon feeds living mussels. Sediments beneath brine may record storms or floods from centuries ago. The bottom is not outside history. It is one of the places where history accumulates. Even so, the animals remain present tense. A shrimp moving through a mussel bed is not a relic. A tube worm extending its plume is not a symbol. A fish hovering over the interface is alive now, responding now, vulnerable now. The deep sea may carry old chemistry, but its communities are not frozen in time. They are active, changing, and contingent. The phrase ocean bottom can suggest a final surface, the end of descent. But a brine pool shows that the bottom has layers beneath it. Fluids come from under the sediment. Salt bodies deform below. Hydrocarbons migrate through buried pathways. Microbial reactions occur inside mud. The visible seafloor is only the upper skin of a deeper system. Sometimes that deeper system reveals itself with bubbles, sometimes with a dark stain, sometimes with carbonate crust, sometimes with a shimmering brine surface, sometimes with animals arranged in ways that make no sense until chemistry is measured. The deep seabed speaks through indirect signs, and much of deep-sea science is learning how to read them without forcing them into a story too quickly. The edge of the pool is a good place to practice that discipline. It tempts metaphor immediately. It looks like a forbidden lake, a dark eye, a portal, a wound, a trap. Some of those images may pass through a viewer's mind, but the narration has to return to what is there: saline fluid, anoxia, methane, sulfide, mussels, microbes, shells, sediment, instruments, and water under pressure. The animal evidence can be subtle. A mussel bed may show different shell sizes, indicating recruitment over time. Dead shells may reveal past positions of the community. Living tissue can show active symbiosis. Isotopes can indicate whether carbon came from methane or other sources. Growth patterns can suggest environmental stability or stress. The shells are not just scenery, they are records. Tubeworm tubes can also persist after the animal dies, leaving structure behind. Other organisms may use abandoned tubes as shelter or substrate. Carbonate blocks can remain after seep flow changes. A former active margin may become a relic community, still visible as shells and crust even after the strongest chemical flow has moved elsewhere. The seafloor contains both active and abandoned edges. Distinguishing active from inactive seep habitat is not always easy from a distance. A shell bed may be living or dead. A white patch may be bacterial mat or exposed mineral. A dark surface may be brine or shadow. Sensors and samples matter because the camera can deceive when light, angle, and sediment combine. The ROV pilot and scientists learn to test impressions carefully. A manipulator claw collecting a mussel must do more than grab an animal. It must avoid crushing surrounding organisms, stirring brine, or losing the sample during ascent. A suction sampler collecting small animals from shell beds must be placed precisely. A water sampler crossing the interface must close at the right moment. These actions are slow because the habitat is fragile at the scale of a tool. Once on deck, samples enter another world of labels, coolers, gloves, microscopes, and chemical bottles. The animal that lived in darkness is examined under lab lights. The water that sat under pressure is transferred into containers. The sediment core is sliced. The datasheet fills with numbers. The distance between the seafloor pool and the ship's lab is enormous, not only in meters, but in conditions. Some organisms survive the trip better than others. Hard shells protect mussels. Worm tubes may remain intact. Microbes can be preserved for genetic work. But behavior is lost unless it was recorded. Position is lost unless it was mapped. Pressure is lost unless specialized chambers maintain it. Every sample carries both information and absence. This is why long, careful observation matters. Before collecting, scientists often watch. They note which animals are alive, how they are arranged, whether bubbles are present, whether the interface is sharp, whether bacterial mats occur upslope or downslope, whether fish avoid the brine, whether crabs feed at the rim. Observation gives the sample its ecological meaning. The deep sea does not reveal itself quickly. A vehicle may spend hours descending and only minutes at a target if weather, power, or schedule limits the dive. A manipulator problem can cost precious time. Poor visibility can obscure the bottom. Navigation errors can place the ROV meters from the intended site, which may be enough to miss a small brine feature entirely. Despite these difficulties, the number of known seeps and brine features has grown as mapping and exploration improve. Multi-beam surveys, autonomous vehicles, submersibles, ROVs, and industry data have all contributed. The ocean is not completely unknown, but it is unevenly known. Some margins are mapped in detail, other regions remain only broadly surveyed. Even in studied areas, small features may wait beyond the last track line. This uneven knowledge should not be treated as failure. It is the nature of studying a vast, deep, dynamic ocean. The camera beam is narrow, but it can be placed thoughtfully. Each dive adds a piece. Each sample tests an interpretation. Each return visit changes a still image into a time series. Deep sea knowledge grows less like a sudden revelation and more like sediment, layer by layer. Brine pools also matter beyond their own boundaries because they test assumptions about habitability. They show that oxygen-free, hypersaline, high-pressure environments can still host active microbial communities. They provide analogs for early Earth conditions and for thinking about life in other salty, icy, or chemically layered environments. The comparison should be made carefully, but the relevance is real. Early Earth did not have animals at brine pool rims, but it did have chemistry, water, minerals, and microbial metabolisms evolving under conditions very different from modern surface habitats. Deep hypersaline anoxic basins give scientists natural laboratories where oxygen is absent and chemical gradients are strong. They do not recreate the origin of life, but they help test how life functions near certain extremes. Astrobiology sometimes enters the conversation because icy moons and subsurface oceans may contain salty water, chemical gradients, and water-rock interactions. It would be easy to exaggerate this into fantasy. The cautious version is stronger. Studying Earth's extreme brines improves understanding of the limits of life, the signatures life leaves, and the kinds of instruments needed to detect biology in difficult environments. Back on Earth, those instruments are still being refined. Chemical sensors become smaller and more precise. Autonomous vehicles can map larger areas. Genomic methods reveal organisms that cannot be cultured easily. Imaging improves. Long-term observatories can watch seep environments over time rather than only during brief expeditions. Each improvement changes the questions scientists can ask. A cabled observatory near methane seeps can measure changes continuously, capturing events a ship might miss. A sudden increase in bubbling, a temperature shift, a current event, or biological response can become part of the record. Brine pools are harder to instrument in some settings because the interface is delicate, but the goal is the same: to see deep sea processes as ongoing rather than as isolated snapshots. The surface weather still controls access. A storm can delay launch. Currents can make navigation difficult. Equipment can fail. The ship must hold position while the vehicle works below. The human schedule meets the ocean schedule, and the ocean is not obligated to cooperate. This practical reality is part of why each clear image of a brine pool is valuable. When the footage finally arrives, it often spreads beyond science. Viewers see an underwater lake and recognize the contradiction immediately. The image is easy to share because it violates expectations without needing explanation. But the explanation deepens the image. It shows why the lake exists, why animals gather at the edge, why the interior can kill, and why the frozen-looking stillness is actually a set of active processes. The best deep-sea images work this way. A glass sponge field is more than delicate architecture when you know how currents bring food through it. A whale fall is more than bones when you know the succession of scavengers and microbes. A vent chimney is more than smoke when you know the chemistry of black smoke or fluid. A brine pool is more than a dark mirror when you know the salt, methane, oxygen, and microbial life around it. The pool edge is also a boundary between ways of seeing. To the camera, it is visual. To the sensor, it is numerical. To the mussel, it is chemical. To the fish, it may be a hazard detected through taste, smell, or bodily stress. To the microbe, it is a metabolic opportunity. No single perspective is complete, but together they make the place more real. Reality includes calm. Not every brine pool scene needs an accident, a dead crab, or a dramatic crossing. Much of the habitat is patient. Animals hold position. Microbes grow without visible movement. The brine's surface trembles and settles. Sediment falls. The pool exists in darkness, while storms pass far above without touching it directly. The drama is in persistence. Persistence, however, is not permanence. Seafloor landslides can disturb brine basins and generate waves along the interface. Fluid flow can increase or decline. Sedimentation can bury parts of a community. Earthquakes can alter pathways. Human disturbance can damage hard grounds. Climate-driven changes in ocean circulation may affect oxygen, temperature, and carbon delivery over longer timescales. The pool is stable only relative to a human visit. Some changes would be difficult to notice without precise measurements. A slight warming of bottom water might alter hydrate stability over time. A change in oxygen could shift microbial processes. A change in current could affect larval delivery. These are not always visible in a single dive. Long-term science is necessary because deep-sea change can be slow, episodic, or hidden until a threshold is crossed. Methane hydrate introduces particular uncertainty because it stores large amounts of methane in sediments globally, but its behavior depends on pressure, temperature, sediment conditions, and local geology. At many seeps, microbes consume methane before it reaches the surface ocean or atmosphere. At the same time, warming bottom waters in some regions could affect hydrate stability. The details are complex, and broad claims require caution. Around an individual brine pool, methane is not an abstract climate number. It is a local chemical source. It feeds microbes. It enters mussel tissue through symbiosis. It may bubble, dissolve, oxidize, or contribute to carbonate formation. The global carbon cycle is built from countless local transformations like this, most of them invisible without instruments. Hydrogen sulfide has a local story too. To humans, it is associated with toxicity and rotten egg smell. Though smell has no meaning at the deep seafloor for us unless a sample reaches air. To bacteria, sulfide can be fuel. To animals, it can be both resource and threat depending on concentration and adaptation. The same molecule changes meaning across bodies. Salt has its own history. The salt beneath the Gulf was once seawater in evaporating basins. Sunlight and climate helped concentrate it. Geological burial hid it. Pressure moved it. Water dissolved it. Brine carried it upward. A mussel bed now grows beside a fluid whose salt began in ancient surface conditions. The deep sea is connected to vanished shallow seas by chemistry. This connection makes the title feel less like a contradiction. Frozen brine pools at the ocean bottom are not simply cold lakes. They are places where ancient salt, modern pressure, methane ice, microbial metabolism, and deep water meet. The frozen part may be hydrate, brinicle ice in polar settings, preserved bodies, or the stillness of dense brine. But the living reality is liquid, active, and carefully bounded. A careful script should not choose one meaning and discard the rest. It should let the title open the door and then clarify. Most deep-sea brine pools are liquid, not frozen solid. Some are near freezing because the deep ocean is cold. Some are warmer due to fluids from below. Methane hydrate nearby may be ice-like. Polar brine can form actual ice structures. The ocean contains all of these without collapsing them into one image. The camera returns to the pool edge because that is where the subject can be held in one frame. The dark brine, the pale shells, the living animals, the sediment, and the machine light are all visible together. A crab crossing the rim would make the danger obvious, but even without that, the boundary is clear. Life has arranged itself around a line. The line is not perfectly sharp forever. Molecular diffusion works against it. Small turbulence works against it. Currents, animal movement, and fluid flow disturb it. But density keeps rebuilding it, and limited mixing allows the brine to remain separate enough for the pool to persist. The surface is therefore a balance between forces that mix and forces that stratify. Stratification is common in the ocean. Warm water can sit above cold water. Fresher water can sit above saltier water. Oxygen-rich water can overlay oxygen-poor water. In fjords, basins, and oxygen-minimum zones, restricted mixing can create strong chemical differences. A brine pool is an intense local version of stratification, compressed into a visible landscape on the seabed. Animals are often good indicators of where stratification becomes biologically meaningful. A fish does not need to know the salinity value to avoid water that stresses its body. A mussel bed does not need to understand methane oxidation to grow where symbionts can feed it. Evolution works through survival and reproduction, not through explanation. Science arrives later to name the gradients the animals have already answered. The ROV's instruments translate those gradients for us. Salinity becomes a number. Oxygen becomes a concentration. Methane becomes a measurement. Temperature becomes a profile. Video becomes mapped observation. The translation is necessary because human senses are poorly suited to a dark, pressurized chemical boundary a kilometer below the surface. That distance is not only vertical, it is experiential. Humans live in air, under one atmosphere of pressure, with sunlight, moderate salinity inside our bodies, and oxygen everywhere we expect it. A brine pool exists in water under high pressure, without sunlight, with salt concentrations that challenge cells and chemistry that can exclude oxygen. To understand it, we have to trust instruments as extensions of sense. Even with instruments, there is room for surprise. A pool may be warmer than expected. A microbial community may contain lineages not previously known from similar habitats. A mussel bed may occupy a position that suggests different flow than the map predicted. A hydrate outcrop may host animals where people expected mostly microbes. The deep sea repeatedly shows that similar processes do not produce identical places. This is why naming a habitat does not finish the work. Calling something a brine pool identifies a major feature, but not its full ecology. The important questions remain. How salty is it? How thick is the interface? What gases are present? How much oxygen reaches the margin? Which microbes dominate? Which animals live there? How long has it existed? How is it changing? The answers require disciplines that do not always speak the same language. Geologists map salt structures and fluid pathways. Chemists measure dissolved compounds. Microbiologists study metabolism and genes. Biologists observe animals and food webs. Oceanographers measure currents and mixing. Engineers build the vehicles that make all of this possible. The pool belongs to all of them because it is made by all those processes at once. In the reference transcript, Deep Time organized the rise and transformation of life. Here, the organizing force is not a geological period, but a physical boundary. Still, time remains present. Salt from ancient basins, methane from buried organic matter, microbes dividing now, animals growing slowly, vehicles visiting briefly. The brine pool is a meeting point of timescales rather than a single moment. A mussel at the edge may not appear impressive beside a giant squid or a whale, but its life is one of the most important stories here. It turns chemical seepage into animal biomass. It builds shell habitat for smaller organisms. It records environmental conditions in its tissues and shell. It links microbes to predators. It makes the invisible chemistry visible as a bed of living bodies. The same is true of microbial mats. They may look like stains or pale films, but they are active communities performing transformations that structure the habitat. Without microbes, methane and sulfide would not become food for most of the visible animals. The deep sea brine pool is not a scene where microbes are background. They are the base of the system. This makes the pool edge one of the places where the old hierarchy of visibility breaks down. Large animals are easier to film, but small organisms may be more important. A crab catches attention because it moves. A fish catches attention because it has eyes. A mat simply lies on sediment, yet it may be processing the chemistry that supports everything the camera is drawn toward. The hidden ocean often works this way. The most important activity is not always the most dramatic. Marine snow falling grain by grain feeds vast areas of bottom. Microbes in sediments regulate carbon and nitrogen. Larvae too small to notice connect distant populations. Symbionts inside animal tissues make survival possible. A brine pool concentrates these hidden processes until the camera can finally see their consequences. There is danger in making every hidden process sound profound, but there is also danger in making it sound ordinary. The balance is to describe it clearly. A dense liquid sits on the seafloor because it is saltier than the water above. It lacks oxygen because it mixes poorly and microbes consume what oxygen enters. Animals avoid the interior but gather at the boundary because microbes turn methane and sulfide into food. That is enough. The quiet facts carry their own weight. A lake can exist under the ocean. A shoreline can form without air. Ice like methane hydrate can sit on the seafloor where pressure holds it stable. Worms can live on that hydrate. Mussels can farm bacteria in their gills. A crab can die by crossing from one water into another. The deep sea does not need exaggeration. The Gulf brine pools are often introduced through their images, but their deeper cause is buried salt. Salt deposits are not rare curiosities in Earth history. They form when seawater is concentrated by evaporation in basins where water enters more easily than it leaves. Over time, minerals precipitate in sequence, leaving layers that can later be buried, folded, pierced, and dissolved by fluids moving through the subsurface. In the Gulf, those salt layers belong to a long tectonic history. The opening of the Atlantic and the development of the Gulf created basins where thick evaporites accumulated. Later sedimentation buried them under kilometers of material. The salt did not simply remain as a flat sheet. Under load, it moved, rose, formed domes and ridges, and helped create pathways for hydrocarbons and saline fluids. Organic matter buried with sediment can be transformed into oil and gas under heat and pressure. Those hydrocarbons migrate upward when pathways exist, often becoming trapped by impermeable layers or salt structures. Some eventually seep naturally to the seafloor. Where methane reaches oxygenated sediment and seawater, microbes respond. Where saline fluids reach depressions, brine pools can form. The same subsurface system can produce both energy source and dense boundary. This connection to oil and gas makes seep habitats scientifically valuable, but politically and economically complicated. Natural seepage shows that hydrocarbons have moved through the seabed long before industrial drilling. At the same time, human extraction changes the stakes because infrastructure, disturbance, and spills can affect habitats that already depend on delicate chemical conditions. Understanding natural seep systems helps separate background processes from human impacts. The animals at the pool edge cannot separate those categories. They live where methane arrives, whether that methane is part of a natural seep field mapped by scientists or part of a region also known to industry. A mussel's symbionts do not care about human terms for resource or reserve. They use the molecule as fuel, and the animal's survival depends on the continued delivery of that chemistry at a tolerable rate. Delivery rate matters. Too little methane or sulfide in the symbiotic system may not support dense life. Too much sulfide or too little oxygen, and the same chemistry can become harmful. The pool edge is not simply the richest place. It is the most balanced place, a narrow band where enough chemical energy meets enough seawater chemistry for animals and microbes to function together. That balance can shift vertically as well as horizontally. Just above the interface, oxygen may be available and salinity tolerable. Within the interface, salinity and chemicals change rapidly. Below it, the brine may be too harsh for most animals. A mussel bed on the rim is therefore living in 3 dimensions, even if it looks spread across a flat edge. Its gills, shell, foot, and symbionts occupy microenvironments separated by centimeters. The brine surface is not a membrane, but it can behave like one in ecological terms. It separates communities. It traps particles. It slows exchange. It creates a place where chemical reactions occur along a plane. If the rub disturbs it, layers fold and swirl, but once disturbance ends, the density difference helps restore the separation. The pool has no skin, yet it has a boundary. Brine can also overflow. If enough dense fluid enters a pool, it may spill across a rim and flow downslope. In footage, such flows can look like smoke or dark water sliding over the bottom. They may follow channels, pool again in lower depressions, or spread thinly across sediment. For animals in the path, an overflow can be lethal, burying or bathing them in water they cannot tolerate. A mussel bed along a stable edge suggests that the pool margin has remained within a tolerable position long enough for settlement and growth. A dead mussel bed drowned by brine suggests a recent change. Shells can therefore indicate the history of the boundary. Living shells show present conditions. Dead shells may mark past conditions. The rim becomes a record of where the pool has been. Some brine pools build raised edges partly because of mineral precipitation and biological accumulation. Shells gather. Carbonate forms. Sediment is trapped. The pool may sit in a crater-like depression or on a mound where fluid flow has altered the seabed. It is easy to imagine the brine simply filling a hole, but many pools are actively constructed by the processes that feed them. The word crater appears in some descriptions, but it does not always mean impact or explosion. It may describe shape rather than origin. A brine pool can occupy a depression associated with fluid expulsion, sediment collapse, salt movement, or carbonate buildup. Seafloor language often borrows familiar landforms, and each borrowed word needs care. The pool's dark color and footage is also partly an optical effect. Dense brine may contain particles or dissolved materials that change how light passes through it. The interface bends light. The bottom may be hidden because the brine is deeper than the camera can see through clearly. The darkness feels symbolic, but it begins as physics, chemistry, and optics. If the ROV lowers a temperature probe into the brine, the numbers may not match the viewer's expectation. Some deep brines are not colder than surrounding water. The hot tub of despair, for example, was warmer than the nearby deep sea, which is why its name includes hot tub. Other brines, especially polar brines, are extremely cold. Frozen brine pools are not a single thermal category. Temperature matters because it affects density, microbial activity, gas solubility, hydrate stability, and animal metabolism. Cold slows many biological processes but can stabilize certain chemical structures. Warmer brine may intensify stress for animals already challenged by salt and low oxygen. In deep habitats, a few degrees can be meaningful because background temperatures are often stable. Stability is relative. A shallow coastal animal may experience daily temperature swings, storms, light cycles, and seasonal change. A deep-sea mussel at a seep may experience a more constant temperature, but a more variable chemical environment. Methane flow can pulse. Brine level can change. Sediment can shift. The deep sea is not always physically quiet in the ways that matter to its residents. The animals at seeps must therefore handle both constancy and unpredictability. They live in darkness and pressure that barely change, but they depend on chemical fluxes that may be patchy. They grow slowly in some cases, but their habitat can be altered by a sudden sediment movement. They are specialized, but specialization ties them to places that may turn against them. Specialization is visible in the mussel's gills. In shallow water relatives, gills function mainly in respiration and feeding. In seep mussels, the gills can also house symbiotic bacteria, changing the organ into a site of chemical agriculture. This does not make the mussel passive. It must regulate water flow, oxygen, sulfide or methane exposure, waste products, and the balance of its internal microbial partners. Symbiosis can be inherited in different ways depending on the animal group. Some symbionts pass directly from parent to offspring. Others are acquired from the environment. For seep animals, environmental acquisition means larvae must not only find a suitable habitat, but also obtain the right microbial partners. The adult community and the microbial environment around it become part of the reproductive landscape. Larval dispersal raises a question of connectivity. If seep habitats are islands, how far apart can they be before populations become isolated? Currents may carry larvae along continental slopes. Chemical cues may help at close range. Long-lived larvae might travel farther, but risk being diluted into unsuitable water. Short-lived larvae may settle more reliably near parents, but fail to colonize distant sites. Genetic studies help answer these questions by comparing populations from different seep fields. Similar genes across distant sites suggest exchange. Strong differences suggest isolation. The patterns can be complex because currents, larval duration, seafloor topography, and habitat age all interact. A brine pool rim may look self-contained, but its population history extends into the water column. Small mobile animals may connect habitats differently. Shrimp, amphipods, and fish can move between patches as adults if distances are manageable and conditions permit. Some may use seeps as feeding stations rather than permanent homes. Others may be more tightly bound to seep chemistry. The community includes residents, commuters, opportunists, predators, scavengers, and microbes that never move far at all. The brine pool interior filters that community. Animals that cannot tolerate it are excluded or killed. Microbes that can tolerate high salt and anoxia may occupy it. The result is not a gradient from life to no life, but a change in what kinds of life can function. Human eyes notice the absence of fish and crabs inside the brine, but the invisible microbial world may remain active. Sampling the brine itself can reveal strong stratification. The top few centimeters may differ from deeper layers. Salinity can increase downward. Oxygen can vanish quickly. Sulfide, methane, metals, ammonium, and other compounds can vary with depth. A single bottle from one level cannot represent the whole pool. The water body is layered, and the layers matter. This is especially true in deep hypersaline anoxic basins. In some Red Sea brines, the interface between normal seawater and brine is a narrow zone where microbial abundance and activity can be high. Above it, conditions resemble deep seawater. Below it, salinity and anoxia become extreme. The interface is not just a transition, it can be the main biological engine. The Red Sea basins also remind us that brine pools can be chemically individual. Some are rich in metals. Some are hot. Some have unusual pH or salinity. Some have microbial communities unlike those in neighboring basins. Because the Red Sea is geologically active and has restricted circulation, its deep brines developed under conditions different from the Gulf of Mexico. Similar words hide different histories. In the Mediterranean, hypersaline anoxic basins relate partly to the Messinian salinity crisis, when large evaporate deposits formed as the Mediterranean's connection to the Atlantic changed. Later dissolution of those evaporates produced dense brines in deep basins. These systems show how ancient climate and sea level history can reappear as modern deep-sea chemistry. The Orca Basin in the Gulf of Mexico represents another kind of large brine-filled depression where dense saline water occupies a basin and creates an anoxic lower layer. Such settings differ from small brine pools at seeps, but share the principle of density-driven separation. The ocean bottom can hold lakes at multiple scales, from small pools to basin-sized brine bodies. In all cases, anoxia changes the rules. Many animals rely on oxygen because aerobic respiration is efficient. Without oxygen, their metabolism fails unless they have special adaptations or can retreat. Microbes have more options. They can use nitrate, sulfate, carbon dioxide, metals, or other compounds in metabolic pathways depending on conditions. This metabolic flexibility makes microbial life central to anoxic systems. Anoxia also slows certain forms of decay. Many familiar decomposers and scavengers need oxygen or animals that do. In an oxygen-free, hypersaline environment, breakdown can be slower or different. Preservation is not perfect or eternal, but it can be strong enough to keep bodies and sedimentary layers recognizable longer than expected. The pool becomes a chemical pause in the normal recycling of remains. That pause is part of the visual power of dead animals in brine. A fish on the bottom appears caught in a state between life and disappearance, but the narration should not linger on horror alone. The body also shows where conditions change, how physiology fails, and how the pool's interior differs from the water above. It is a measurement written in tissue. The same pool that preserves a fish may feed a mussel bed at the edge. This is not contradiction if the gradients are understood. The interior and the margin are different habitats. A few centimeters can separate them. Life and death are not distributed evenly across the feature. They follow chemistry. Chemistry also helps explain why bacterial mats can be so sharply placed. White mats often appear where sulfide reaches oxygenated water. Orange, red, or dark films can reflect different microbial groups, mineral particles, or chemical conditions. Without samples, color alone can mislead, but it gives researchers a visual clue about where to measure. The ROV's lights make those colors available to humans, but many deep-sea animals may rely more on chemical and mechanical cues. A crab can taste or smell dissolved substances. Fish can detect flow and odor. Shrimp may respond to surfaces and chemical traces. The edge is likely sensed before it is seen, because natural light is absent, and the interface itself may be visually invisible to animals without artificial illumination. Vision still matters for some deep-sea species. Many fish have large eyes adapted to low light or to bioluminescence. But at the seafloor, chemical sensing can be crucial. A methane seep has a smell in the chemical sense, a plume of dissolved compounds carried by currents. Animals that locate food or habitat may follow these gradients in ways difficult for humans to imagine, because our noses do not work underwater at depth. Currents shape those gradients. A weak bottom current can carry sulfide or methane traces in one direction, elongating bacterial mats or influencing where animals settle. A stronger current can erode sediment, expose carbonate, or deliver oxygenated water to a margin. The pool surface may be still, but the water above it is not motionless. Flow also supplies oxygen to the animals at the rim. Chemosynthesis needs reduced chemicals, but many symbiotic animals also require oxygen. Too little current, and the boundary may become oxygen poor. Too much disturbance, and the interface may mix, or the animals may experience stress. Again, the habitat depends on balance, not simply on abundance of one ingredient. Balance can be seen in the spacing of animals. Dense mussels may occupy one band. Tube worms may cluster where sediment chemistry suits their buried roots. Bacterial mats may occupy softer areas. Crabs may concentrate along paths where prey is accessible. Fish may hover slightly above structure. The community is a map of tolerances. If a pool margin is filmed repeatedly, changes in those bands can reveal ecological shifts. A new mussel cohort may appear as small shells among older ones. A bacterial mat may expand after increased seepage. Dead shells may accumulate where brine advanced. Predators may respond to increased prey. Over time, the edge can be read almost like a shoreline affected by rising water, erosion, and colonization. Deep sea time series are rare, but valuable. They turn habitats from nouns into verbs. Instead of saying a brine pool is a feature, scientists can ask how it moves, breathes, overflows, stabilizes, or reorganizes. Instead of saying a mussel bed exists, they can ask how it recruits, ages, declines, or recovers. Technology is slowly making that possible. Autonomous underwater vehicles can map broad areas before ROV dives. High-resolution photomosaics can record an entire pool rim in detail. Repeat surveys can be compared pixel by pixel. Chemical sensors can be left in place for limited periods. Machine learning may help classify animals in long video records, though human expertise remains essential for interpretation. Even simple lights have changed knowledge. Before submersibles and ROVs, much deep-sea understanding came from dredges, trawls, and indirect measurements. Those tools were powerful, but destructive and context-poor. Seeing the animals alive at seeps changed interpretations. A mussel bed in a trawl might suggest abundance. A mussel bed around a brine pool shows why the abundance is where it is. The history of deep-sea exploration is full of such corrections. Animals once thought rare may be common in specific habitats. Behaviors inferred from dead specimens may look different in life. Structures assumed geological may be partly biological. Empty-looking mud may contain microbial processes that instruments reveal. Each new tool does not simply add detail, it changes the frame of what counts as evidence. Brine pools are especially dependent on in situ observation because their defining feature is a fluid boundary. Bring brine to the surface, and it is still salty water, but the pool shape, interface behavior, pressure conditions, and biological arrangement are gone. The lake exists as a lake only in place. Remove it from place, and you have samples, not the habitat itself. This is why the narration keeps returning to the camera beam. The beam is limited, but it preserves the relationship between parts. It shows the dark brine beside the shells, the crab beside the rim, the sampler entering the water, the ripples after thruster movement. It keeps the pool from becoming only a chemistry table. The table matters too. Salinity values show why the brine sinks. Oxygen measurements show why animals cannot breathe inside it. Methane concentrations show how microbes and symbionts are fueled. Sulfide values show both toxicity and energy. Temperature tells whether hydrate may be stable and whether the pool differs from ambient conditions. Numbers discipline the image. A good deep-sea script has to hold both forms of evidence without letting either dominate for too long. Too much image and the habitat becomes a mood. Too much chemistry and the animals disappear. Brine pools are powerful because the image and chemistry fit together so closely. The line the viewer sees is the line the instruments measure and the animals obey. There is also a tactile quality, even though no human hand can safely touch the pool at depth. The ROV arm can place a sampler into brine and the surface deforms. A corer can slide into sediment near the rim. A shell can break under the manipulator. Fine mud can billow. These textures remind us that the deep sea is physical, not merely distant. Pressure makes direct human contact impossible without a submersible. In a human-occupied vehicle, researchers look through thick acrylic or glass viewports surrounded by a pressure hull. In an ROV operation, the people remain on the ship and send a machine instead. Either way, the human body is separated from the habitat by engineering, because the deep ocean is not a place we can enter unprotected. This separation influences knowledge. Humans bring lights, cameras, clocks, sample jars, and categories. The animals bring bodies shaped by pressure, salt, darkness, and chemical flow. The interaction is brief. A dive may be enough to describe a scene, but not enough to know every seasonal pattern, every larval event, every microbial shift. The limits are part of the truth. The most honest deep-sea narration often says what is known and then stops before invention. Scientists know that brine pools can be toxic to many animals. They know that microbes and symbionts support life at margins. They know that salt density creates the surface. They may not know exactly how often a certain species crosses the boundary, how old a specific pool is, or how its chemistry varies through every season. Uncertainty can be visual too. The camera sees a fish near the pool and we do not know whether it is hunting, resting, investigating the ROV light, or simply passing through. A crab at the edge may be feeding on microbial film, scavenging, or testing the boundary. Without repeated observation or sampling, behavior should be described cautiously. The animal is present. Its exact intention is not always available. This restraint does not make the scene less engaging. It makes it more faithful. The deep sea is not a theater where every movement has a dramatic meaning. Much of life is routine. A fish conserving energy may be more accurate than a fish confronting death. A crab stepping around brine may be avoiding physiological stress rather than acting out a story of fear. Energy conservation is a major deep-sea theme. Many animals move slowly or wait because food is limited and metabolism may be adapted to cold. At seep habitats, local food can be more abundant, but energy still matters. Predators do not waste effort without reason. Scavengers respond to opportunities. Sessile animals invest in structures and symbioses rather than chasing food. The brine pool's margin can concentrate opportunities enough to attract mobile animals from surrounding areas. A fish may learn that mussel beds hold prey. A crab may patrol shell edges. Amphipods may gather on dead material. The pool interior kills some animals, and their remains may become a resource where accessible. The hazard and the food web are not fully separate. Scavenging in or near brine is complicated by toxicity. A dead fish inside the brine may be inaccessible to ordinary scavengers if entering the pool is dangerous. Material at the edge may be used more readily. Microbes adapted to anoxic high salt conditions may process remains differently. The pool changes not only who dies, but who can feed on death. This can affect nutrient cycling locally. In ordinary deep sediment, a carcass might be quickly visited by scavengers and broken down. In brine, the pathway may be slower and more microbial. Nutrients may remain trapped or released differently. Small-scale chemistry influences how energy moves through the community. The pool also has acoustic and hydrodynamic quietness. Dense brine can damp some mixing, and the interface can hold particles. But the surrounding deep sea still carries sound from currents, animals, ships, and geological activity. The idea of total silence is another simplification. The deep ocean is dark to human eyes, but not empty of vibration, flow, or chemical signal. Human sound may reach deep habitats. Ship engines, seismic surveys, and industrial activity can travel through water. The effects on seep communities are not always obvious and may vary by animal. This is an area where caution is appropriate. Brine pools are not isolated from the broader oceans simply because they are deep. They are also not isolated from surface climate. Surface productivity affects marine snow. Ocean circulation affects oxygen and temperature at depth. Climate history affects salt formation and basin chemistry over geological time. Modern warming may affect some hydrate systems depending on depth and region. The connections are complex, but the deep sea is part of Earth's changing system. At the same time, one should avoid making every brine pool a climate warning. The habitat has its own biology and geology beyond human concern. Methane seeps matter to carbon cycling, but they are also homes for animals. Hydrate stability matters to climate models, but hydrate outcrops also support microbes and worms. The science is strongest when multiple meanings can coexist without forcing the place into a single message. The deep sea often becomes a screen for human imagination because it is hard to see. Brine pools are especially vulnerable to this because they look impossible at first. The answer is not to remove wonder, but to anchor wonder in mechanism. Salt made the water dense. Density made the surface. Anoxia made the interior hostile. Methane and sulfide fed microbes. Microbes fed animals. The chain is clear and still remarkable. A viewer may remember the image as a lake under the ocean. A scientist may remember the conductivity profile. A microbiologist may remember the interface sample. A geologist may remember the salt structure. An ecologist may remember the mussel distribution. The habitat gives each observer a different entry point, and a complete account needs all of them. The idea of a frozen lake at the bottom of the sea sounds like myth until the pieces are separated. Frozen in appearance, the still brine surface. Frozen in chemistry, methane hydrate locked as a solid under pressure. Frozen in preservation, bodies and sediment layers protected by anoxia. Frozen in polar formation, brinicles growing around sinking brine. None of these alone defines every brine pool, but together they explain why the title feels intuitively right. The brine itself remains liquid because salt depresses freezing. This is why roads can be treated with salt in winter, and why sea ice rejects brine as it forms. The same principle appears in a far stranger setting, when concentrated brine stays fluid under conditions that would freeze fresher water. The ocean's freezing behavior is not a single temperature. It depends on salt, pressure, and composition. Methane hydrate complicates the visual field because it can look like ordinary ice while containing gas. If broken or warmed under lower pressure, it can release methane and water. On the seafloor, hydrate can be part of the landscape, hidden under sediment or exposed in patches. It may coexist with seep communities, but its stability zone depends on local pressure and temperature. The animals living on hydrate outcrops are not living on magic ice. They are using a surface where microbes can grow and where methane is available. The hydrate may be hard, irregular, and chemically active at its boundary. Worms moving over it are part of a microbial-animal relationship, not evidence that the ice itself is food in a simple sense. In the same way, Animals at brine rims are not feeding on salt. Salt shapes the pool, but methane, sulfide, microbes, and organic matter support the food web. A mussel bed exists because bacteria can turn chemical energy into biomass. The salt sets the stage, draws the boundary, and excludes competitors, but it is not the fuel that feeds the community. The stage can be narrow. In some footage, the living band around a pool appears only a short distance wide. Inside is dark brine. Outside is less productive sediment. Between them is the place where conditions match the needs of symbiotic animals. This thinness should change how the viewer understands vulnerability. A small shift in the pool edge can matter. Many deep-sea habitats have such narrow tolerances. Corals need currents and hard substrate, but can be damaged by sedimentation. Vent animals need chemical flow, but not lethal heat. Oxygen minimum zone animals need low oxygen adaptations, but still have limits. Brine pool animals need seep chemistry and tolerable salinity. Survival is often a matter of position. Position is also what cameras can record well. A photomosaic of a pool rim can show exactly where mussels, mats, carbonate, and dead shells occur. Over time, repeated mosaics can quantify changes. This kind of visual ecology turns the cinematic image into data without removing its atmospheric power. The same photograph can be beautiful and measurable. Measurement becomes especially important when comparing pools. A viewer might assume all underwater lakes are alike because they share a visual form. Scientists compare salinity, depth, temperature, area, interface thickness, methane concentration, sulfide, animal composition, microbial genes, and geological setting. The comparison reveals diversity within the category. Some pools have names because humans remember named places better than coordinates. Hot Tub of Despair is memorable, perhaps too memorable, because it emphasizes danger and warmth. But the name also helps draw attention to a real feature that scientists studied with water samples, mapping, and visual surveys. A dramatic name can open a door, but the scientific work has to walk through it. Names can also bias perception. A pool called Despair may make every animal near it seem doomed. A pool called a lake may make it seem familiar. A pool called a seep feature may make it sound technical and dry. The narration has to move among these terms without being trapped by any one of them. The place is dangerous, familiar-looking, and technical at the same time. The animals do not require names to function, but naming them helps humans notice differences. A crab is not simply a crab if its species has particular tolerances, diet, and distribution. A mussel is not just a shell if it hosts methane-oxidizing symbionts. A worm is not just a tube if its body plan is built around bacteria. Taxonomy gives texture to the community. At the same time, overloading the script with names can obscure the scene. The reference style explains terms when they matter and returns to the bigger movement. For brine pools, the important biological roles are clear: symbiotic mussels, tube worms, microbial mats, mobile scavengers, predators, and animals that avoid or fail to avoid the brine. Names should support those roles, not replace them. A deep-sea animal's body often makes sense only when its environment is included. The mussel's gills make sense with methane. The tube worm's plume makes sense with sulfide and oxygen. The crab's cautious movement makes sense with a lethal boundary. The fish's hovering makes sense with energy conservation and prey concentration. Remove the habitat, and the body becomes an oddity. Restore the habitat, and the body becomes practical. This is especially true for preserved specimens. A dead seep mussel in a museum drawer cannot show the shimmering brine surface beside it. A tube worm tube cannot show the chemical gradient that fed its symbionts. A water sample cannot show the animals avoiding the pool. The specimen is essential, but it needs the scene in order to recover its full meaning. The same is true for the scene-needing specimens. Video alone cannot reveal all symbionts, genes, isotopes, growth rates, or chemical concentrations. A beautiful dive without sampling may inspire, but it may not answer the hard questions. A destructive sample without video may measure, but lose ecology. The strongest deep-sea science pairs site with material. A brine pool is therefore a place where several kinds of patients meet. Geological patience made the salt and pathways. Microbial patience builds mats and processes methane. Animal patience grows mussel beds and tubeworm fields. Scientific patience maps, measures, samples, and returns. The ROV footage appears immediate, but it rests on slow systems. When the ROV light grazes a mussel bed, the shells cast small shadows. Those shadows are one of the few signs of three-dimensional structure in an otherwise dark field. Between shells, water moves differently. Particles settle. Larvae find crevices. Small animals hide. A bed of mussels is not only a population, it is habitat architecture. Habitat architecture can outlast the living animals. Dead shells remain as hard substrate. Tubes remain as shelter. Carbonate remains as ledge. This means a seep community can influence future communities even after chemistry changes. The physical remains of life become part of the next habitat, just as coral skeletons build reefs in shallow water, though the organisms and processes differ. The analogy to reefs should be limited, but useful. A mussel bed around a brine pool is not a coral reef, but it can increase local complexity, provide surfaces, and create niches for other organisms. Complexity matters in the deep sea because flat, soft sediment offers fewer attachment points and hiding places. Where structure appears, animal diversity can increase. Carbonate crust adds another layer of structure. It can create cracks, overhangs, and hard surfaces. Corals or sponges may attach where conditions allow. Fish may use relief. Crabs may move along edges. The chemistry that precipitates carbonate, therefore, changes not only the water, but also the geometry of the seafloor. Geometry affects flow. A raised rim can deflect currents over a pool. A mound can create eddies. A depression can trap dense brine. Shell beds can slow near bottom water. These flow effects influence oxygen delivery, particle deposition, larval settlement, and chemical dispersion. The pool is not just chemistry sitting on shape. Chemistry and shape modify each other. If a landslide enters a brine basin, the interface can respond like a lake surface, generating internal waves. Studies of subsea brine pools have shown that underwater landslides can disturb these dense layers. This is another reminder that fluid surfaces do not require air above them. A boundary between two waters can carry waves if the density contrast is strong enough. Such internal waves would be mostly invisible without instruments or lucky timing. The ROV might see the interface rising and falling, but more often, the event would be reconstructed from deposits, models, or measurements. Much of the deep sea's motion is episodic. A quiet dive can pass through a place shaped by rare events that happened long before the vehicle arrived. Sediment around brine pools can record those events. Layers may show turbidites from underwater flows, chemical precipitates, biological remains, or disturbances. In anoxic basins, the lack of burrowing can preserve laminations. To a sedimentologist, the quiet mud beneath brine is not blank. It is a document, written slowly and protected by conditions that exclude many animals. The lack of burrowing animals is itself ecologically meaningful. Bioturbation, the mixing of sediment by organisms, is common in many seafloor habitats. It oxygenates sediment, redistributes organic matter, and blurs layers. In brine interiors where animals cannot live, that mixing is reduced or absent. Chemistry changes the physical record by changing who can disturb it. The interface above such sediment can hold its own microbial communities. Cells may concentrate where nutrients and electron acceptors meet. Viral communities may influence microbial mortality and gene transfer. These details are far beyond what the ROV camera can show, but they are part of the living pool. The visible shoreline is only the upper scale of a much smaller biological world. Viruses in deep sea brines may sound like a side note, but they are part of microbial ecology. They can shape which microbes dominate, move genetic material, and influence nutrient cycling by lysing cells. In an extreme habitat, viral communities may also reveal how life interacts under stress. The deep sea's smallest agents can affect its largest patterns. The more closely scientists look, the less the pool resembles a simple dead zone. It is deadly to many animals, but not dead in a universal sense. It excludes much visible life, but supports microbial complexity. It traps bodies, but feeds margins. It is extreme, but organized. The phrase dead zone is useful only if carefully defined for oxygen-dependent animals, not for all life. This distinction matters because the deep sea has often been misunderstood as empty or lifeless. Brine pools demonstrate something more precise. Some areas within them are hostile to most animals. Around them, life can be dense. Within them, microbial life may be highly adapted. The correct picture is not empty darkness, but sharp biological sorting. Sharp sorting is common wherever environmental gradients are steep. On a rocky shore, barnacles, mussels, algae, and snails occupy zones according to immersion, drying, waves, and predation. Around a brine pool, zones form according to salinity, oxygen, methane, sulfide, substrate, and flow. The deep-sea shoreline is different, but zonation as a biological principle is familiar. This familiarity helps without making the habitat ordinary. A shore under the ocean is still strange, but it is not beyond understanding. The same ecological logic that organizes life elsewhere operates under pressure and darkness, using different materials. Life responds to gradients because gradients create both limits and opportunities. A viewer who returns to the footage after learning this may see more. The white mussels are not decoration. The empty dark interior is not simply ominous. The ripple is not just cinematic. The crab's path is not random. The boundary is a working part of the ecosystem, and every organism near it has a relationship to that boundary. Some relationships are direct. A symbiotic mussel needs methane. A sulfide oxidizing mat needs sulfide and oxygen. A fish avoids brine. A crab feeds on mussels or associated animals. Other relationships are indirect. A coral may attach to carbonate produced by seep processes. A predator may visit because prey gather there. A larva may settle because microbial films signal suitable substrate. Indirect relationships are often harder to prove. Scientists may propose them based on patterns, but then need experiments, samples, or repeated observations. Does a certain animal prefer active seep carbonate or any hard surface? Does a larva respond to methane-associated microbes or to shell texture? Does a predator rely heavily on seep prey or only opportunistically use them? The questions are specific because ecosystems are built from specifics. Specificity also helps avoid exaggeration. Not every brine pool is surrounded by spectacular life. Not every seep has the same animals. Not every hydrate outcrop hosts visible worms. Not every animal near a pool is specialized for it. Some are generalists taking advantage of structure. Some are visitors. Some may be there by accident. The deep sea is diverse enough without forcing every observation into a grand pattern. One animal at the edge can still carry the whole system if described carefully. Imagine a mussel half buried in shell hash, its valves slightly open, drawing water across gills filled with bacteria. Around it, salinity is tolerable but influenced by brine. Methane diffuses from below. Oxygen arrives from above. The animal is a living intersection of the pool's physics, chemistry, and biology. Imagine a tube worm plume extending into weak current. The tube anchors the animal in sediment. Inside its body, symbionts process chemicals. Outside, the plume exchanges gases with the water. The worm does not need to move across the seafloor because the habitat moves through it as dissolved molecules. Its stillness is not inactivity. It is a different strategy. Imagine a crab at the rim, one claw raised slightly as sediment shifts under its legs. It may be feeding, exploring, or avoiding the brine. Its hard shell protects it from predators, and pressure does not crush it because internal fluids balance the environment. But chemistry can still defeat it. Armor helps against teeth and claws, not against water that cannot support respiration. Imagine the brine itself without animals visible. A dark surface under black water, rippling from an unseen current, holding particles at its boundary. For a human mind, the absence invites story. For a scientist, the absence invites measurement. Salinity, oxygen, methane, sulfide, temperature, density, the empty-looking surface is full of variables. Variables connect to equations, but the script does not need to become mathematical. It only needs to show cause and consequence. More salt means more density. More density means less mixing. Less mixing means oxygen can be depleted. No oxygen excludes most animals. Chemical energy at the boundary supports microbes. Microbes support animals. The logic is simple enough to follow and complex enough to occupy a career. Career-long study matters because deep-sea habitats rarely yield final answers. Each expedition returns with new samples and new complications. A microbe appears that does not fit previous expectations. A pool's chemistry differs from a neighboring pool. A model predicts one flow path, and mapping reveals another. Science advances by adjusting to these mismatches. This adjustment is visible in language. Early descriptions may emphasize death and extremity. Later work adds symbiosis, microbial diversity, geochemistry, physical oceanography, and carbon cycling. The pool does not change because language improves, but human understanding becomes more layered. The underwater lake becomes an ecosystem, a chemical reactor, a geological feature, and an archive. Archives require preservation, but preservation is selective. A sediment core from an anoxic basin may record fine layers, but it may not show animals excluded from the brine. A dead fish in the pool shows an accident or boundary crossing, not necessarily common behavior. A mussel shell bed shows successful settlement, but not every failed larva. The record always has a bias. Recognizing bias is part of respecting the deep sea. The ocean does not arrange evidence for convenience. It preserves some things and erases others. Soft bodies vanish. Rare events may leave strong traces. Common processes may be invisible. A camera can miss what happens outside the beam. A sample can overrepresent what is easy to collect. The truth is assembled from imperfect pieces. This is why the voiceover should feel careful, not hesitant. Carefulness is not lack of confidence. It is confidence placed where evidence supports it. A brine pool is dense hypersaline water in a seafloor depression. That can be said firmly. The exact behavior of every fish near every pool cannot. Strong narration knows where to stand and where to leave space. Leaving space is appropriate for the ending too, but before the camera leaves, the system deserves one more pass from below to above. In the sediment, microbes process methane and sulfate. In the brine, salt and anoxia exclude most animals. At the interface, reactions and microbial communities intensify. On the rim, mussels, tube worms, mats, and mobile animals form a living edge. Above, the deep ocean carries currents, larvae, and falling snow. Above that are kilometers of water, then weather, ships, satellites, and ordinary human days. The pool is not separate from those layers, but it is hidden by them. A person can cross the Gulf in an airplane and never suspect that a salt lake lies below the flight path. The deep sea remains close in planetary terms and remote in experience. Remote places can still be affected by distant actions. Carbon dioxide changes ocean chemistry. Fishing gear can touch deep bottoms. Resource extraction can alter sediments. Plastic and pollutants can sink. Noise can travel. The brine pool is specialized, but not immune to the world above. Its depth protects it from some disturbances and exposes it to others that move through water, sediment, or human industry. The most practical response is knowledge. Map the features. Understand the communities. Identify vulnerable structures. Study recovery. Separate natural variability from disturbance. Protect rare habitats where possible. None of this requires turning the pool into a moral symbol. It requires treating it as real, specific, and worth knowing before it is changed. Knowledge also changes the viewer. After seeing a brine pool, the ocean bottom no longer feels like a flat endpoint. It can hold lakes, rivers, ice like hydrates, chemical gardens, animal cities, archives, and traps. The darkness becomes less like absence and more like a place where light has not been brought yet. The hidden ocean gains detail. Detail is the opposite of generic mystery. Mystery says the deep is unknown. Detail says a crab stood here, a mussel bed rimmed this edge, a sensor measured this salinity, methane rose through this sediment, salt came from ancient evaporation, and a camera watched particles spread across a liquid boundary. Detail does not remove wonder. It gives wonder a body. The body of the pool is water altered by salt. The body of the community is shell, tube, mat, claw, fin, and microbe. The body of the evidence is video, sample, map, number, and preserved specimen. All of these bodies are partial. Together they allow humans to approach a place we cannot inhabit. The approach should remain slow to the end. The ROV does not rush over the surface. It holds position, corrects drift, and lets the interface settle in the frame. In that stillness, the title becomes clearer. Frozen does not mean lifeless. It does not mean simple ice. It means a set of conditions where water, salt, gas, cold, pressure, and life have found forms unfamiliar at the surface. A final scientific correction is worth keeping. Brine pools are not rare because the ocean lacks salt. The ocean is full of salt. They are rare because the conditions needed to concentrate brine, release it, and trap it on the seafloor are specific. There must be a source of extra salt, a pathway, a depression or basin, limited mixing, and often a seep system that adds chemical energy. When those conditions come together, the result can be visually startling, but it is not accidental. It is the predictable outcome of physical laws acting in a complicated earth. Salt dissolves. Dense fluid sinks. Pressure stabilizes gases in unusual forms. Microbes use chemical gradients. Animals gather where microbes make food. The underwater lake is strange because the circumstances are uncommon, not because the rules have failed. The rules are what make it possible to trust the image. Without them, the pool would be only a curiosity. With them, it becomes a place that can be studied, compared, revisited, and understood more deeply over time. Every measurement adds another edge to the picture, like light moving across a shoreline that was always there, but not yet visible. Even the word shoreline changes after this. A shoreline can exist without air. It can be made of shell and carbonate at the bottom of the sea. It can separate oxygenated water from anoxic brine. It can be crowded with animals that do not know the sun. It can be filmed by a machine connected to a ship by cable. It can be ancient in cause and active in the present. The next time the camera finds such a line, the scene will not feel like a trick. The dark surface will still be beautiful, but it will also be legible. The viewer will know that the pool is heavy with salt, that the edge is chemically alive, that the animals are positioned with precision, and that the apparent stillness is maintained by forces moving too slowly or too invisibly to see. This is the deep sea at its most instructive. It shows that an environment can be hostile and productive at the same time. It shows that life is not evenly spread, but concentrated by gradients. It shows that ancient geology can feed modern communities. It shows that a place can look frozen while remaining chemically active. It shows that darkness is not a lack of story, but a condition under which different stories occur. There is no need to make the pool larger than it is. A small brine pond on the seafloor is enough. Its edge contains geology, physics, chemistry, microbes, animals, technology, uncertainty, and time. The camera only has to hover, and the facts begin to accumulate. A long descent to a brine pool begins with hardware that looks almost industrial on deck. Cables are checked, connectors are sealed, thrusters are tested, and sample boxes are arranged where the manipulator can reach them. The deep sea scene that will later feel quiet and remote begins in noise, salt spray, diesel, helmets, gloves, and people confirming that every part of the vehicle can survive the pressure below. Once the ROV is lifted over the side, the ocean closes around it quickly. The surface chop that seemed large from the deck becomes a thin moving ceiling above the cameras. Sunlight flashes on bubbles and then softens into blue. Instruments begin logging. The descent is mechanical and patient, a controlled fall through water that changes from weather-driven surface to layered ocean. For a long time, there may be nothing in the main camera except particles. That emptiness can mislead a viewer who expects constant animals. The water column is immense, and the camera sees only a small cone. A jelly may pass outside the lights. A fish may avoid the vehicle. A copepod may be too small to notice. Life is present, but not always visible in the frame. Depth readings provide the first sense of scale. 100 meters, 200, 500, 1,000. Pressure increases steadily, even though the water looks similar on camera. At these depths, human lungs would collapse without protection, but many animals live without any drama because their bodies have no air spaces to crush, and their chemistry is adapted to the surrounding pressure. Near the bottom, the camera begins to catch suspended sediment. The altimeter counts down. The lights reveal a pale plain, sometimes marked by burrows, tracks, shell fragments, or small animals that freeze in the beam. The vehicle slows. Thrusters adjust. After the long vertical transit, the world becomes horizontal, a seafloor where centimeters and meters suddenly matter. A brine pool may not be obvious immediately. The ROV may follow a slope, cross ordinary mud, pass a carbonate block, and then find a change in texture. A bacterial mat appears. Shells become more abundant. A fish hovers near the bottom. The scientists in the control room may see these signs before the pool itself, because seep habitats often announce themselves through life and minerals before the main fluid body comes into view. Approach too quickly, and the thrusters can erase the clarity of the scene. Fine sediment rises in clouds. The interface blurs. Animals move away. Good pilots learn to move gently near such features, using small corrections and watching the water as much as the bottom. The ROV is powerful, but the best observations often come from restraint. The first close view of the interface can be disorienting. The camera sees a horizon where no horizon should be. The brine surface may reflect light unevenly, and the water above it may appear to tremble. Particles settle onto the boundary and slide. The pool does not look like ordinary mud or rock. It looks like a separate substance occupying the same world as everything else. A conductivity probe confirms what the eye suspects. As it enters the brine, the reading changes sharply. Salinity rises. Oxygen may fall. Temperature may shift. The sensor gives the scene a numerical border. A viewer sees a dark surface. The instrument records a density step. Together, they make the underwater lake real in two different ways. The manipulator may collect water from just above the interface first. This sample represents the world of fish and crabs, the water that bathes the mussel bed and carries oxygen. Then, a second sample may be taken within the interface, the thin zone of transition. A third may come from deeper brine. These three bottles can differ dramatically, despite being collected within the same small frame of video. Back in the lab, those bottles may be handled as if they are fragile layers of a place. Chemical analysis can reveal methane, sulfide, chloride, sulfate, dissolved inorganic carbon, metals, nutrients, and pH. Microbial DNA can reveal communities that occupy different depths. The pool that looked like one dark body becomes stratified into habitats stacked inside a few meters of water. This stacking is why careless mixing matters. If the interface is stirred before sampling, the layers blend and the strongest gradients may be lost. The same problem occurs in many aquatic systems, from lakes with anoxic bottoms to oxygen minimum zones and stratified fjords. Boundaries are scientifically valuable because they are sharp. Disturbing them can erase the evidence one came to measure. The animals at the rim also depend on those gradients remaining usable. Too much mixing could bring harsh brine upward. Too little exchange could reduce oxygen supply. In nature, currents and density maintain a shifting balance. The animal community is built around that balance, and the ROV must avoid becoming a temporary storm. A suction sampler may be used to collect small mobile animals from the shell bed. Amphipods, small shrimps, worms, and snails can vanish into crevices before a manipulator claw can reach them. Suction allows a more delicate capture, though it still alters the microhabitat. Each collected animal carries information about the food web that larger camera targets cannot provide alone. A push core near the rim can capture sediment layers, microbes, pore water, and small infauna. The core is pushed straight down and capped before retrieval. Inside it, the top layer may show oxygenated sediment, while deeper sections reveal sulfide or methane-rich zones. The core is a vertical cross-section through processes that the surface view only hints at. The simplest object in the sample box may be a mussel. Its shell can be measured. Its tissue can be analyzed for stable isotopes, symbionts, and contaminants. Its gill can be examined under a microscope. Its age may be estimated in some cases. What looked like one animal at the rim becomes a record of diet, chemistry, growth, and habitat history. Stable isotopes are especially useful because they can show where carbon and nitrogen in tissues came from. Animals relying heavily on methane-based production can carry chemical signatures different from animals feeding mainly on surface-derived organic matter. This helps scientists determine how much of the community is fueled by seep chemistry and how much by ordinary deep-sea food sources. The answer is often not all one or the other. Some animals at seeps may feed directly or indirectly on chemosynthetic production. Others may eat a mixture of seep-derived and background organic material. Predators may integrate both sources by eating prey from different habitats. Food webs are not clean diagrams. They are networks with many routes for carbon to move. A brine pool rim is therefore not sealed from the surrounding sea. Marine snow still falls there. Currents still deliver particles. Mobile animals still move in and out. The seep adds energy, but it does not erase the broader ocean. The community is built from both local chemical production and regional deep-sea context. This matters for interpreting abundance. A dense mussel bed may suggest that seep energy dominates locally, but predators visiting the bed may also feed elsewhere. A fish filmed near the pool may not be dependent on it. A scavenger may arrive because the pool traps animals, but also use other carcasses on the slope. Ecological dependence is a matter of degree. The brine interior is even more separate in some ways and connected in others. It resists mixing, but dissolved substances still diffuse across the interface. Particles still settle onto it. Microbes at the boundary can process materials from both sides. The pool is isolated enough to develop distinct chemistry, but not so isolated that it has no exchange with the surrounding ocean. This partial exchange is common in nature. Total isolation is rare. Total mixing is also rare. Most habitats exist between those extremes, exchanging some materials and retaining others. Brine pools are striking because the balance is visible. The dark surface shows retention. The living rim shows exchange. The deep ocean's cold helps preserve gradients by reducing turbulence in some settings and slowing certain processes, but it is not a guarantee of stillness. Bottom currents can be energetic. Internal waves can move along density layers. Storms at the surface can influence deep water indirectly through currents and pressure changes, especially along margins. A calm-looking bottom can be hydrodynamically active over time. Seep animals experience this activity through flow. A tube worm plume bends. Mussel siphons orient. Bacterial mats fray at edges. Sediment ripples. Shell hash accumulates in low-energy pockets. The direction and strength of water movement influence where oxygen and chemicals meet, making current a hidden architect of the brine pool community. A chemical plume from a seep can extend beyond the visible pool. Dissolved methane, sulfide, or other compounds may travel with currents, diluted but detectable. Animals may use such plumes as cues. Instruments can follow them to locate seep sources. The pool itself is only the most visible part of a larger chemical field. In some searches, scientists do not know exactly where the brine pool is before the dive. They may use sonar anomalies, previous records, oil slicks at the surface, bubble detections, or seafloor maps to choose a target. The ROV then performs a visual search, reading signs from the bottom. Exploration becomes a combination of prediction and recognition. Recognition improves with experience. A pale bacterial mat may suggest sulfide. A cluster of chemosynthetic mussels may suggest methane. Carbonate crust may suggest long-term seepage. Dark fluid in a depression may suggest brine. But each sign can have exceptions. Good observers remain alert to both pattern and surprise. Surprise is one reason these habitats continue to matter after decades of study. A new species may appear in a sample. A known species may be found at a new depth. A pool may show unusual chemistry. A microbial genome may reveal an unexpected metabolism. The deep sea is not unknown in a simple sense, but it remains capable of revising assumptions. The revision can be small and still important. Perhaps a brine interface is thinner than expected. Perhaps oxygen penetrates farther in one pool because currents are stronger. Perhaps a mussel bed survives closer to the brine than models predicted. These details may not become popular headlines, but they refine how scientists understand the limits of life. Limits are not fixed lines drawn once for all organisms. They are species-specific, life-stage-specific, and context-specific. An adult mussel may tolerate conditions a larva cannot. A microbe may thrive where a crab dies. A fish may survive a brief dip into stressful water, but not prolonged exposure. The boundary between possible and impossible shifts with body, duration, and adaptation. Duration is crucial. Some animals can endure low oxygen for short periods. Others can regulate internal salt balance within limits. A crab touching the brine and retreating is different from a crab sinking into it. A fish passing through a thin layer is different from a fish trapped below the interface. Exposure time turns a harsh condition into either a warning or a death sentence. This helps explain why the pool edge can be investigated by animals without constant catastrophe. A mobile animal may test the boundary, encounter stress, and withdraw. It may learn through sensation or instinctive response. The brine is dangerous, but danger does not mean every contact is immediately fatal. The severity depends on the pool, the animal, and the exposure. The most dramatic footage often shows animals that did not retreat in time, but ordinary footage may show animals simply staying near the margin. Both matter. Death reveals the boundary. Avoidance reveals adaptation. Feeding reveals opportunity. A complete account needs all three and should not let the dramatic event define the whole habitat. Deep sea channels and slopes can carry carcasses, wood, and plant debris from shallower waters. These materials may arrive near brine pools and add temporary food. Woodfalls can support their own communities of borers, microbes, and associated animals. The seep community exists amid this broader range of irregular resources, some chemical, some biological, some geological. The surrounding mud may contain burrowing animals that are not obvious on camera. Polychaetes, bivalves, small crustaceans, and meiofauna can live within sediment, processing organic matter and oxygen. Near brine, their distribution may change sharply. A core taken outside the pool can be compared with one near the rim and one inside to show how salinity and oxygen structure hidden fauna. Meiofauna are often overlooked because they are tiny, but they can be abundant and ecologically important. Nematodes, copepods, foraminiferans, and other small organisms occupy sediment spaces between grains. Some tolerate low oxygen or high salinity better than larger animals. Their patterns can reveal environmental stress at scales the ROV camera cannot resolve. Foraminiferans, with their shells or tests, can also leave records in sediment. Different species tolerate different conditions, and their remains may indicate past oxygenation or salinity. In layered brine basin sediments, microfossils can help reconstruct environmental history. The smallest shells become part of the archive. The archive beneath brine may preserve events from the surrounding land as well. Dust storms, floods, earthquakes, and climate shifts can alter sediment input. In places where layers remain undisturbed, these signals may be read long after the event. A deep sea brine basin can therefore hold traces of surface environments without ever seeing sunlight. This vertical connection between surface and bottom is one of the ocean's great organizing facts. Surface plankton feed deep animals through sinking particles. Land-derived dust can fertilize surface waters or settle into deep sediments. River floods can send material to slopes and basins. Ice formation can produce dense brine that contributes to deep water. The bottom is hidden, but not disconnected. Polar brine formation makes the connection especially physical. Sea ice grows at the surface, rejects salt, and produces dense water that sinks. On large scales, this contributes to water mass formation. On small scales, it creates brine drainage channels and sometimes brinicles. The surface freezing of seawater can therefore influence the deep and near-bottom ocean through density. Brinicles have a different visual drama from Gulf brine pools. They grow downward as ice tubes, pale against dark polar water, fed by brine draining from sea ice. Around them, the water can be cold enough that the tube continues to extend while the brine flows inside. When the flow reaches the seafloor, it can spread and freeze or stress animals in its path. This phenomenon is sometimes called the icy finger of death in popular media, but that phrase should be handled carefully. It captures the visible danger to slow animals, but it can make the process sound like a predator. The brinicle is not hunting. It is a physical structure formed by freezing, salt rejection, and density-driven flow. Animals caught by it are affected by chemistry and cold, not intention. The comparison between brinicles and deep brine pools clarifies the title. Both involve brine sinking because it is dense. Both can create bottom hazards. Both can produce striking boundaries. But brinicles are ice structures beneath sea ice, generally in shallow polar waters, while deep brine pools in places like the Gulf often form from dissolved buried salt and remain liquid. The shared word brine covers different worlds. Methane hydrate is a third world. It is frozen-looking, pressure-stabilized, and often associated with methane seeps. It may occur near brine systems because hydrocarbon migration and salt tectonics can be part of the same regional geology. Yet hydrate is solid and brine is liquid. A careful narration keeps them linked but distinct. If a piece of hydrate is disturbed at depth, bubbles may escape or the surface may change. Scientists collecting hydrate must use specialized tools if they want to preserve it under pressure. Otherwise, it can dissociate during ascent. This makes hydrate research technically demanding. The substance that looks like ice is stable only because the deep sea supplies the right conditions. Gas hydrate has attracted attention as an energy resource, a climate factor, and a geological hazard. But within seep ecosystems, it is also substrate and chemical source. Worms, microbes, and other organisms associated with hydrate outcrops remind us that geological materials can become biological surfaces. The seafloor does not divide neatly into living and non-living components. Salt crusts, carbonate blocks, shells, hydrate, bacterial mats, and sediment all interact as habitat. A shrimp may walk over carbonate, feed on microbes, hide between shells, and avoid brine. A worm may live in sediment influenced by methane and sulfide. A mussel may attach to hard material built by previous seep activity. The community is assembled from many materials at once. The apparent simplicity of the pool is therefore deceptive. From a distance, it is a dark patch with a rim. Up close, it is layers, flows, reactions, surfaces, bodies, and histories. A script can move from distance to detail in the same way the ROV does, first seeing the lake, then the shoreline, then the shells, then the microbes and chemistry implied by them. At times, the narration should also move back outward. The pool is one feature on a continental margin. The margin is one part of an ocean basin. The basin is part of the planet's circulation and carbon cycle. Deep-sea storytelling works best when it can shift scale without losing the animal or object in the frame. A mussel shell can lead to Jurassic salt, then return to the shell. The reference transcript often moves from concrete image to broad process, and back again. That rhythm suits brine pools because the habitat itself is a bridge between the small and the vast. A single ripple at the interface is caused by local disturbance, governed by density physics, and made possible by geological salt. Small image, broad explanation, specific return. A natural transition might begin with the ROV lights bending at the brine surface, then explain density, then return to a fish avoiding the edge. Another might begin with a mussel bed, explain chemosynthetic symbiosis, then return to the shell pavement. This keeps the narration from becoming either a lecture or a sequence of disconnected images. The emotional intensity should remain restrained because the subject already carries enough strangeness. A lake under the sea does not need to be called impossible. It is possible, and that is the point. A pool that kills fish does not need to be described as evil. It is a harsh chemical environment. A microbial mat does not need mystical language. It is living chemistry made visible. Restraint also respects the animals. They are not props in a scene of danger. The crab near the rim is a working organism. The mussel bed is a community of feeding animals. The fish over the pool is navigating its habitat. The microbes are performing metabolism. Their lives are not about human awe, even if awe is a reasonable human response. There is an ethical clarity in treating deep-sea animals as ordinary in their own terms. The more extreme the habitat seems to us, the more important it is to describe the organisms as adapted rather than monstrous. A tube worm with no adult gut is not incomplete. It is specialized. A mussel dependent on bacteria is not strange for effect. It is a successful animal in a chemical ecosystem. Success can be local. An animal may be perfectly suited to one seep field and absent from another. A microbe may thrive in one brine chemistry and fail in a neighboring basin. Evolution does not produce universal solutions. It produces workable arrangements under particular pressures. Brine pools make that specificity visible because conditions change so sharply across short distances. The term extremophile is often used for microbes in brines, and it is useful, but it is also relative. Extreme for a human cell may be normal for a halophilic archaeon. Extreme for a fish may be manageable for a microbe. The word describes distance from human comfort as much as biological difficulty. A good script can use it while remembering that each organism experiences its environment from its own baseline. Pressure is similar. Humans call the deep sea high pressure because our bodies evolved at the surface. Deep animals are not constantly fighting pressure. Their molecules and membranes function under it. Bring them up, and the surface becomes the extreme environment. A brine pool mussel is not living in a hostile world from its own perspective, as long as the chemistry remains within its tolerances. That inversion is useful. The deep sea is not alien to its residents. It is home. The brine edge is not a nightmare to a mussel if methane and oxygen arrive properly. The darkness is not darkness in a human emotional sense, it is the normal light condition. The pool is strange to us because we are visitors, and our categories were built at the surface. Even the idea of bottom has a surface bias. We think of bottom as an end, a final floor. But for seep systems, the bottom is a boundary between ocean and subsurface. Fluids move upward. Chemicals cross. Microbes live within sediment. The brine pool is a window into the underworld of the seabed, not the end of the ocean's structure. Below the visible sediment, pore spaces hold water, gas, salts, and microbes. Reactions occur without light. Methane may be consumed before it reaches open water. Sulfate from seawater may penetrate downward and meet methane rising upward. At that sulfate-methane transition, microbial processes can drive carbonate formation and shape the chemistry that eventually appears at the surface. These buried reactions are hard to film. The camera sees their products: bubbles, mats, carbonate, brine, animals. Scientists infer the subsurface from cores, chemical profiles, heat flow, seismic data, and models. The visible brine pool is only the expression of a larger hidden plumbing system. Seismic imaging can reveal salt bodies and subsurface structures at scales far beyond NOAA's view. Industry and academic surveys map layers, faults, domes, and pathways. These images are not cinematic in the same way as animal footage, but they show why the pool appears where it does. The shoreline on the seafloor is connected to shapes buried deep below. The connection between hidden structures and visible animals is one of the most satisfying parts of seep ecology. A salt body deforms sediment. A pathway opens. Brine and methane move upward. Microbes use the chemicals. Mussels settle. Crabs arrive. A fish hovers in the rove light. Geology becomes biology through a chain of causes. This chain can break at any point. If no pathway exists, chemistry stays buried. If brine disperses without pooling, no underwater lake forms. If methane flux is too low, dense symbiotic communities may not develop. If salinity is too extreme at the margin, animals may be excluded. The habitat is the result of conditions aligning, not just one factor. That alignment makes brine pools valuable for teaching how ecosystems are built. They are not defined by one charismatic animal. They are defined by relationships among salt, water, gas, microbes, animals, sediment, and pressure. The subject is a habitat, and the main character is a boundary. A boundary can be difficult to make emotionally engaging because it has no face. Brine pools solve that by making the boundary visible. The interface is the face of the system. It ripples, reflects, bends light, traps particles, and divides animals from danger. It gives the camera something to look at, while the narration explains what cannot be seen. The best moment may be when the rove light catches both the surface and the rim animals at once. In that single view, the viewer sees why the habitat matters. The dark brine is not separate from the mussels. The mussels are not separate from the microbes. The microbes are not separate from the methane. The methane is not separate from buried geology. The line holds the whole chain. Even after thousands of words, the essential image remains simple, heavy water lying under heavy water. It is simple enough for a child to grasp and complex enough for oceanographers, geochemists, microbiologists, and ecologists to study for decades. That combination gives the topic its strength. The script should eventually narrow again, because an ending cannot hold every fact at once. It should return to one pool, one camera, one edge. After traveling through salt tectonics, polar ice, methane hydrate, microbes, animals, sampling, and uncertainty, the viewer needs the concrete place again. The rove is still there. The brine is still dark. The edge is still alive. A final pass through the science can begin with the simplest experiment anyone can imagine. Put salty water beneath fresher water, if the salty water is dense enough, and it will stay low. The ocean performs that experiment at vast scale with temperature and salinity, but a brine pool performs it in miniature on the seabed, where the difference is sharp enough for a camera to see. The experiment becomes more complicated because seawater is not just water and salt. It contains dissolved gases, carbonate chemistry, nutrients, metals, organic molecules, microbes, and particles. When brine is concentrated through buried salt or ice rejection, those other components may also change. The pool is not merely stronger seawater. It is a chemically distinct water body with its own reactions. The interface acts as the main exchange surface. Oxygen from above can enter only slowly. Methane, sulfide, ammonium, and other compounds from below can rise or diffuse upward. Microbes position themselves where reactions are favorable. If the balance shifts, the community shifts. A thin layer can therefore regulate the movement of matter between the pool and the open deep sea. In ordinary ocean water, oxygen allows animals and many microbes to break down organic matter efficiently. In anoxic brine, different pathways dominate: sulfate reduction, methanogenesis, fermentation, and other anaerobic processes can become important depending on chemistry. These pathways often produce compounds that would be toxic or reactive in oxygenated water, adding to the pool's distinct character. The smell of sulfide is familiar at the surface near rotten eggs or some marshes, but in the deep sea, smell belongs to chemistry rather than human sensation. An animal does not need a human nose to detect dissolved sulfide or other cues. Chemoreception in water can guide behavior, warn against danger, or lead an organism toward food. The pool edge is sensed chemically before it becomes visible to us. Fish near brine pools may use taste and smell along with lateral lines and vision. A lateral line detects water motion and vibration, helping a fish hold position or respond to approaching animals. Near an interface, subtle flow patterns may matter. The fish is not simply floating in dark water. It is reading a field of chemical and mechanical information. Crabs read the bottom through legs, claws, antennae, and chemical receptors. Their movement can look hesitant because each step gathers information. Around a brine pool, that information may include substrate firmness, food traces, dissolved chemicals, and the edge of intolerable water. A crab that survives is not brave. It is well tuned to local conditions. Shrimp may swarm around some seep features, especially where microbial production or detritus offers food. Their bodies are small and quick compared with mussels and tube worms. They can exploit spaces between shell and crust, flee predators, and move with currents. The camera may catch them as flashes, but their abundance can indicate how much energy is moving through the lower levels of the food web. Snails and limpets can graze on microbial films. Polychaete worms can burrow, crawl, filter, or graze depending on species. Amphipods can scavenge. These smaller animals turn microbial productivity and organic debris into prey for larger animals. Without them, the connection between mat and fish would be weaker. The food web depends on many bodies too small to dominate the frame. Bivalves other than mussels can also occur in seep habitats, including clams with their own symbiotic relationships. Some live partly buried in sediment where sulfide or methane is available. Their shells may be less visually dramatic than a dense mussel rim, but they add to the range of strategies animals use to tap chemical energy without sunlight. The phrase chemical energy can sound abstract, so it helps to compare it to the green world above. In surface ecosystems, plants and algae use sunlight to build organic matter. At seeps, microbes use chemical reactions to build organic matter. In both cases, other organisms depend on primary producers. The difference is the energy source and the environment in which production occurs. This comparison should not imply that chemosynthesis replaces photosynthesis across the whole deep sea. Most deep-sea life still depends heavily on surface production. Chemosynthetic ecosystems are local concentrations, important and spectacular, but unevenly distributed. The brine pool's edge is rich because it is special, not because the entire ocean bottom is equally fueled from below. The contrast with surrounding sediment is therefore part of the story. Move away from the pool, and the density of large animals often falls. Shell beds thin out, bacterial mats disappear, carbonate gives way to soft mud. The camera returns to a more typical deep-sea background, where food arrives mostly from above, and animals may be spaced far apart. That surrounding mud is not empty either. It contains burrows, tracks, small animals, microbial communities, and chemical processes in pore water. The brine pool is spectacular because it concentrates life and creates a visible boundary, but the quieter mudplain has its own ecology. The deep sea should not be reduced to only its dramatic features. Still, dramatic features are useful because they reveal principles. A brine pool reveals density stratification. A vent reveals geochemical energy. A whale fall reveals succession and scavenging. A seamount reveals current concentration. A canyon reveals transport from shelf to deep basin. Each feature makes one aspect of the hidden ocean easier to see. For brine pools, the principle is the power of dissolved material to reshape habitat. Salt is invisible once dissolved, but it changes water's behavior enough to build a lake. Methane is invisible once dissolved, but it feeds microbes enough to build animal communities. Oxygen is invisible, but its absence can decide which animals live or die. The most important parts of the scene cannot be seen directly. This is why instruments are not optional. Human vision is drawn to edges, animals, and textures, but sensors reveal the invisible gradients that explain them. Without conductivity, the pool might be only a visual oddity. Without oxygen measurements, its lethality would be guessed from bodies. Without methane and sulfide data, the animal community would seem disconnected from its fuel. A carefully planned dive may therefore move in stages. First, map the pool. Second, photograph the rim. Third, measure the water column above and within the brine. Fourth, sample animals and microbes. Fifth, collect sediment cores. Sixth, leave enough of the habitat intact that future observations remain meaningful. Exploration and restraint have to work together. The desire to collect everything is understandable because ship time is precious. But deep-sea habitats cannot be treated as unlimited. A mussel bed may take years to recover from disturbance. A small pool margin may be altered by a few careless manipulator moves. Scientific sampling is necessary, but it should be proportional and deliberate. The animal's slow growth in some deep habitats adds weight to that caution. Cold temperatures, limited food, and long lifespans can mean that recovery is slow. Not every seep animal grows slowly, and not every community is fragile in the same way, but many deep-sea systems do not rebound on human schedules. The first rule is to know what is there before assuming it can replace itself quickly. This caution is becoming more important as the deep ocean attracts more human attention. Energy resources, minerals, cables, fisheries, and carbon storage proposals all involve the seafloor in different ways. Brine pools and seeps may be small in area, but they can be biologically and scientifically valuable. Their rarity makes mapping and protection more important. Protection begins with recognizing that an apparently small patch of bottom can contain an entire specialized system. A pool a few tens of meters across may hold microbial lineages, symbiotic animals, chemical gradients, and geological records not found in surrounding mud. Size alone is a poor measure of significance. The same is true for time. A brief RAW clip may show a place that took millions of years of geological history to make. Ancient salt, buried carbon, slow tectonic deformation, and modern seepage all converge in a frame lasting seconds. The shortness of the video should not shrink the timescale of the feature. When the script approaches its end, it should not claim that brine pools solve the mystery of the deep sea. They solve some questions and open others. They show how underwater lakes form, how life uses chemical gradients, and how danger and productivity can share an edge. They do not explain every seep, every microbe, every animal movement, or every future change. The unresolved parts are not failures. They are invitations to return. Another dive, another core, another genetic survey, another long-term sensor, another map. The deep ocean is learned through repetition because no single descent can hold it all. Even a clear brine pool with a famous name remains only partly known. The final image can therefore be calm rather than conclusive. The ROV does not need to witness a death or discovery in the last minute. It can simply hover over the rim while particles fall and the brine holds its surface. The science has already given the image weight. The quiet is enough. At that point, the viewer understands why the animals are where they are. The mussels mark a usable gradient. The bacterial mats mark chemistry. The empty interior marks exclusion. The ripples mark density. The dark pool marks salt, anoxia, and time. The camera is no longer looking at a mystery without language. It is looking at a habitat with rules. Those rules do not make the place less strange. They make it stranger in a more durable way. A fantasy can be dismissed after the image fades. A real underwater lake, fed by ancient salt and methane, bordered by animals farming bacteria in darkness, remains strange because it is true. The ocean bottom has room for such places. The script should leave the pool in its own world. No final moral needs to be forced onto it. The brine does not teach a lesson. It shows a set of conditions and the life arranged around them. That is enough for a deep sea ending. A physical place, a clear image, a remaining boundary. One more detail belongs near the end because it is easy to overlook. Brine pools are not separate from the animals that never enter them. A fish avoiding the brine still participates in the habitat by feeding along its margin. A crab that circles the rim without crossing still responds to the pool's presence. The boundary organizes behavior even when it is not crossed. Avoidance is a form of ecological knowledge, though not conscious knowledge in the human sense. Bodies learn through evolution. Sensory systems detect stress. Muscles turn away. Individuals that respond correctly survive more often than those that do not. Over many generations, the community around the pool becomes shaped by the ability to use the edge without being taken by it. The same principle applies to microbes, but at a chemical scale. Microbial populations increase where their metabolism works and decline where it does not. Their distribution may look like a mat, a colored layer, or nothing visible at all. They do not choose the interface as animals choose a path, but their growth maps the same boundary in another language. Language is always a translation here. Lake, shoreline, ice, pool, death, oasis, frozen, hot tub. Each word gives the mind a handle, and each one risks simplifying the place. A lake under the ocean is not a lake in the ordinary sense. Methane ice is not the ice in a glass. A death pool is also a microbial habitat. The narration has to use familiar words while gently correcting them. Correction does not need to be cold. It can be part of the wonder. The reason the brine lake is real is more interesting than the first impression. The reason frozen can mean hydrate, brinicle, preservation, and stillness is more interesting than a simple block of ice. The reason animals survive at the edge is more interesting than the idea that the deep sea is only dangerous. The deep sea becomes more compelling as it becomes more specific. A vague darkness can hold any imagined monster, but a brine pool holds actual mussels, actual tube worms, actual microbes, actual salt, actual methane, and actual instruments hovering in pressure. The specific world is smaller than fantasy and much more durable. This durability matters for a long-form voiceover. Over many minutes, the subject has to keep unfolding without becoming inflated. Brine pools allow that because every layer leads to another layer: visual surface, density, salt history, seep chemistry, microbial metabolism, animal symbiosis, preservation, sampling, polar comparison, hydrate, carbon cycle, uncertainty, and return to the rim. The pace should remain steady because the habitat itself is steady. The camera does not rush. The brine does not boil or roar. The animals do not perform for the viewer. The strongest movement is often a slow ripple, a drifting particle, a crab leg placed carefully on sediment, or a manipulator closing around a sample. The narration can trust those small movements. If the final seconds hold on the interface, the viewer now knows why it matters. That thin shimmer is not only an image, it is a density boundary, an oxygen boundary, a salinity boundary, a microbial habitat, a trap, a feeding edge, a sampling challenge, and a record of ancient geology rising into the present ocean. A line of water becomes the center of the story. The ocean above remains immense, but the pool does not need to compete with that immensity. Its scale is local and complete. A few meters of brine can contain enough complexity to occupy scientists for years. A narrow rim can support an entire community. A dark patch on the seabed can connect surface evaporation millions of years ago to a living crab in the camera beam. When the machine leaves, the pool returns to conditions that existed before the lights arrived. No narration remains there. No music. No surface language. Only water layered over water, animals at the edge, microbes in the chemistry, and salt holding the dark fluid low against the seafloor. A final scientific humility remains. There are brine pools no camera has visited yet, small seeps hidden on slopes, dense fluids lying in depressions beyond map tracks, microbial layers that no sample has recovered, and animal behaviors that happen between expeditions. The ocean bottom is not completely unknown, but it is incompletely witnessed. That distinction matters because it keeps the subject grounded while leaving room for discovery. Future dives may find pools with different chemistry, rims dominated by different animals, hydrate structures in unexpected positions, or sediment records that preserve events not yet recognized. They may also find ordinary-looking features that become important only after analysis. In the deep sea, discovery is not always a sudden creature in the light. Sometimes it is a number on a sensor, a gene sequence in a sample, or a layer in a core. The brine pool teaches patience because its most important processes are slow or hidden. Salt moved over millions of years. Methane rose through sediment, grain by grain. Microbes built communities at chemical boundaries. Mussels settled and grew along a narrow margin. A camera arrived for a few hours and turned all of that into a visible scene. The short human visit depends on a long non-human history. As the ROV moves away from the pool, the edge may disappear quickly. The beam returns to sediment with scattered shells and faint tracks. The dark surface falls behind. Without coordinates, a person could pass over ordinary-looking mud nearby and never know a hypersaline lake sat just beyond the light. The deep sea is full of such abrupt transitions. The vehicle may climb a few meters and look back. From above, the pool becomes an oval or irregular patch, edged by pale life. The interface reflects the lights faintly. A fish passes over, but does not descend. The manipulator arm folds inward. The pilots prepare for the next transect. Science moves on, not because the pool is finished, but because ship time is limited. Later, the footage will be reviewed. Scientists may pause at frames where the interface rippled, where a particular animal crossed the edge, where the sampler entered, where bubbles escaped. They may compare the video with chemical data and maps. A moment that looked atmospheric becomes quantitative. A shimmer becomes evidence of density. A shell bed becomes habitat distribution. The final understanding remains partial, but not empty. We know enough to say that brine pools are among the ocean's most extreme seafloor habitats. We know they form when dense saline fluids collect in depressions. We know many are tied to cold seeps and subsurface salt. We know their interiors can be anoxic and lethal to many animals. We know their margins can support chemosynthetic life. We also know that each pool must be approached as its own place. The Gulf, the Red Sea, the Mediterranean, polar ice margins, and hydrate-bearing slopes do not all tell the same story. They share physical principles, but their depths, temperatures, salinities, chemistries, and communities differ. Good science keeps both the pattern and the exception in view. The ending returns to the camera, because the camera is how most people first meet the pool. A rectangle of light moves across a dark seabed. The edge appears. Animals gather along a line they did not draw, but have learned through evolution to use. Below that line, dense brine holds its own shape. Above it, the ocean continues, cold and pressurized, carrying snow, larvae, odors, and traces of life. For a while, the machine hovers, and two oceans share the same frame. One is the surrounding sea, oxygenated enough for fish and crabs, connected to currents, snow, and the wider deep. The other is the brine, darker and heavier, salt-rich, oxygen-poor, preserving, excluding, feeding microbes at its boundary. The difference between them is thin enough to ripple and strong enough to organize a community. When the rub finally turns away, the pool does not vanish. It simply leaves the beam. The mussels remain at the rim. The microbes continue in layers too small to see. The brine's surface settles after the thrusters. Methane moves upward through sediment. Salt keeps the dense water low. Marine snow falls through black water and disappears into a landscape that is not empty, not frozen in the simple sense, and not fully known.